Hello.
Yeah,
Okay, okay, fine. No, I'm just checking the sound. Okay, fine. Thank you.
am I audible clearly?
Yes, yes, you're audible clearly. Good morning, all. We will start our today's webinar within few minutes. We'll wait for few more minutes for more participants to join. Thank you. A very good morning and welcome everyone once again in the 2-day national webinar. Today is the second day for this webinar. Tomorrow, the day started with inaugural session and with keynote speech by Professor Mondol on the history of the journey of botany and traditional medicine of India, followed by two plenary speech by Dr. Yogesh Mishra on cadmium stress and recovery in Anamina species, and by Professor Krishnendu Acharya on the present and future of organic farming in India. After that, we had witnessed some path-breaking work through poster session 1, where 18 wonderful posters were presented by the participant, and the day 1 was concluded by our honorable convener and head of the department, Dr. Ashok Bhattacharya, with his concluding remarks and summarization for the day. So, here we are once again to begin a new day with new enthusiasm, and we will start today's webinar with post session 2. Just a little housekeeping before we get started. During the poster session, please keep your microphone in off mode uh, apart from the presenter, and after the session there will be a questionnaire portion. A small recap about the rules and regulations of the poster session. Every participant will was allotted with 7-minute timing, 4 minutes for the presentation and 3-minute interaction session. I will call name of every participant one by one. They have to share their own screen and will present his or her poster. If there is any technical glitch, please come to us without any hesitation, and then only we will share your poster from our side. So, welcome everyone once again and a very good morning. The first poster for today's session is from Mr. Parth Patel from Department of Botany, University, School of Science, Gujarat University. Mr. Parth Patel, are you there? Parth Patel, or any of the co-author of this poster? Like Jyoti Chauhan, Miss Jyoti Chauhan, Miss Nilam Kalal, Mr. Dipesh Gupta, Mr. Miss Suhani Parekh, anyone? I guess they are not present with us. So we'll move to our next poster, that is from Shramana Ghosh, Miss Shramana Ghosh, from
Yes, yes.
from Environmental Biology Laboratory, Department of Botany, Vishwabharati Shantiniketan. And I will notify Ms. Sandeepa Shaha from Department of Conservation Biology to keep her poster ready, as she is the next one. So Shramana Ghosh, Ms. Shramana Ghosh, you can please present your poster. Thank you.
So I would like to know if the slide is visible.
Till now, no.
Is it visible now?
No, 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 no. Should we share from our side?
Yes, but I have to send the recent copy to you then now because
Haven't you submitted it earlier?
yes, I had, but that there was few changes. So can I please again send the copy first?
Uh, okay, then we will move to our next participant. In the meantime, you send the file to us, okay?
Okay, okay, okay. I'm sending it right now.
Okay, thank you.
Thank you.
So the next participant is Miss Shundipa Shaha from Department of Conservation Biology, Durgapur
Yes,
Government College.
yes, sir, I'm present.
Okay. Can you please share your screen?
Yes, sir, I'll try.
Shundipa.
Yes, sir.
So, are you sharing your screen?
Sir, is it visible?
No, it is not visible. Have you sent us your e-poster?
Yes, sir.
So, should we share from our side?
Yes, sir. Okay.
Okay. Mr. Anish Bhattacharya, can you please help me with sharing the poster of Ms. Shundipa Shaha?
Yeah, I'm doing it.
Okay, thank you.
Is it visible now?
Yes, sir.
Okay. Best of luck.
Okay. Thank you. Good morning, everyone. Myself Sandeepa Shaha, and I am very thankful to the organizer for giving me the opportunity to present a poster on flowering plant diversity of Biharinath Hill in Bankura district, West Bengal. A floristic study refers to the listing of all plants of a particular area, and diversity measures the variety and variability of the listed plant. It is also very important for conservation perspective. The present survey-based study involves the qualitative diversity of flora of Biharinath Hill and its surrounding area. The base of the hill is a popular tourist spot because the environment offers a pollution-free green surroundings. It is very important to understand the biodiversity of an area so that local people and also the tourists can be aware of the richness of species and their responsibility to maintain the diversity. My study site is Biharinath Hill and its surrounding area. It is the tallest hill of Bankura district in the Indian state of West Bengal and one of the dense forest areas of the district. Uh, so-
Mr. Anish Bhattacharya, excuse me, sorry,
okay,
Shandipa.
okay, no problem.
Yeah,
Okay,
you can
thank you.
continue.
Okay. It is 447.8-meter high and the area is having a gradual descent from the Chotanagpur plateau. This is a residual hill and almost spherical in shape. Soil of this area is basically lateritic and Bhabur formation is found in the Damodar riverine zone. The vegetation of this region is partially similar to dry peninsular sal forest and northern dry mixed deciduous forest dominated by Soria robusta, Terminalia arjuna, Butea monosperma, etc. The study was conducted from March 2021 to March 2022 and prepared a checklist for flowering plant species around the Biharinath Hill. Now the methodology. Random survey was made throughout the hill slope during 2021. Plants growing in the hill slope and of the surrounding were collected, tagged and recorded in field notebook. Specimens were then processed and identified through conventional technique in Conservation Biology Department of Durgapur Government College. Preliminary identification of specimens were made using literature like Bengal plants, flora of Bankura district, flora of West Bengal, and on the website, the plant list was consulted for updating species name. Now the result, the study deals with 128 flowering plant species belonging to 110 genera. Uh, and the study deals with 128 flowering plant species belonging to the genera 110 genera covering 47 flowering plant families. Among them, 94% terrestrial vegetation, 5% aquatic, and only 1% parasitic plants were found. The vegetation represented 39% herbs, 31% trees, 16% shrub, 8% climber, and 6% lianas. Non-flowering plants in this region are very least, but the number of flowering plants is substantial. Some artificial ponds or water bodies also exist, but aquatic plant diversity is moderate. Among 47 Angiosperm family, Asteraceae and Fabaceae represented the majority. Now the conclusion, the present survey is the first step to explore vegetation and it is found that the flowering plant diversity of Biharinath Hill is very rich. This is the small part of my pilot survey for ethnobotanical study on Biharinath Hill and adjoining area of Saltora block. The vegetation extended up to Shusunia Hill and the area harboring a large number of Santal villagers. Generally, the villagers depend on the vegetation. The dependency of these indigenous people on the existing vegetation of the area may be further explored through extensive ethnobotanical survey. Lastly, I am very grateful to all of my teacher of Conservation Biology Department for their proper guidance to conduct the survey. Thank you.
Thank you so much, Ms. Shah. Now the session is open for interaction.
Okay.
Any questions for Ms. Shah? From any participant? Any question for the presenter?
I have a question. So, for application part, what we will say? I mean, you have done the conservation study, you have studied the biodiversity of that area, Biharinath Hills. So, what is your future plan?
My future plans to ethnobotanical survey. The area is inhabited of many indigenous people, mainly the Santal tribes. So this area my main work is focused on the dependency of those indigenous people on this vegetation.
And how you will do that ethnobotanical study or survey?
I have not started yet. This is my first application, a preliminary study to enlist the plant, and then I study the vegetation first, and after that I will start my next ethnobotanical survey.
Okay, thank you.
Shudhipa, I have one question. You have studied the forested diversity. So which plant species is dominant in this particular area?
Mainly Fabaceae and Asteraceae family.
That's the family. I want some names.
From Fabaceae family, the Butea monosperma, Butea superba, these are dominating. The Shoria robusta from others, and Asteraceae are also.
Which
Acacia.
Asteraceous plants are dominated there?
Asteraceae.
Asteraceae.
Mycenia mercantha is there, and lots of.
Regarding the conservation strategies, is there any endangered or exotic or different categories, different other categories of plants? Do you find any other types of plant?
Exotic plants is Lantana camara is present there. And
Regarding conservation,
yes, ma'am.
and if there are any endangered or rare plants are there?
No, I cannot find.
Actually, madam is asking you that the any rare species, whether any rare species is there or not, because you are trying to do your work in conservation aspect also, and whenever you find any work in the conservation aspects, then the rare species should be identified at first in a particular area. So if you don't find any rare species in that particular area, then why so we utter for the word conservation? That's why madam is saying, I think. So
Yes,
am I right?
Yes.
Yes.
And my main focus on the ethnobotanical study.
Regarding ethnobotanical study, do you study on this particular tribe or total community? Specific community are there, na? They use the plant. Which community you studied?
No, I have not studied yet, but I will explore the total vegetation first, and then I studied the ethnobotanical.
Okay, this is your preliminary work. Is
Yes,
it?
yes, ma'am.
Okay.
Any other question for our presenter? No. So I guess we can move to our next presenter, Ms. Shramana Ghosh. Are you there?
Yes, I'm there.
Yes. Can you see the screen?
Yes, can you please magnify the screen a bit?
Okay. Just a second. Is that okay?
Yes, that's okay.
Okay, you can start.
Okay, so good morning everybody. I'm Shramana Ghosh from Department of Botany, Vishwabharati Shantiniketan. I'm presenting my e-poster on eucalyptus pollen allergy current scenario. The work was done under the supervision of Professor Kashinath Bhattacharya. An allergy is a chronic condition involving an abnormal reaction to an ordinary harmless substance called an allergen, and various allergenic diseases encounter more than 25% of the world population. And in India, the number certainly is more than 10%. Eucalyptus being one of the potential aeroallergens for Australian population has also found to be significant for at least 30% of allergy sufferers of Indian population as well. So in the present paper, an attempt has been made to validate the data and to identify the specific allergen present in them. As we can see in the figure 1, a Google map showing the red bordered area is the main study site that includes the Shonajuri tourist spot, the meteorological department, Shurul area, and a part of Durgapur Wildlife Sanctuary, which is well known for eucalyptus cultivations. So I have divided my entire work into 4 components. One is pollen morphological study through scanning electron microscopy. Another is aerobiological survey and air sampling using Durgapur 7-ray volumetric sampler. The third component is allergy
Can you see
related
the screen?
hospitalization survey.
Sorry to interrupt you. Can you see the screen?
No.
Just a second.
Yes, now I can see the screen. Okay. So as I was saying, the 4th component is the biochemical study. So can you please scroll down a bit? The result, but yes, so in the result we can see figure 2 is the inflorescence of eucalyptus, and the figure 3 is a scanning electron microscopic image showing the pollen grain is oblate with a tricolporate and rugulate exile, colpivir angular, and paracin colpate. So an aerobiological survey was also done. Can you please scroll it down to the Yes, an aerobiological survey was done for a period of 2 years from September 2018 to August 2020, and a correlation was made between the monthly percentage contribution of total pollen grains and monthly percentage contribution of eucalyptus pollen grains, and it was found that the 2 peak season for eucalyptus pollen grains would be between September to November, somewhat in early December, and another would be between March to April. We also did an hospitalization survey, as you can see in figure 5. Can you please scroll the screen a bit up? No, a bit more. To the top, actually. Yes, so the figure 5 is the hospitalization survey data showing a total 39,781 patients, out of which 21.4%, that is 8,500, are admitted associated to allergy related problem, and of that 55.2%, that would be 4,691 patients were admitted related to respiratory tract infection related problems. So what we did, we further made a correlation graph and understood that some of the respiratory tract infection related hospitalization admission coincides within the peak season of eucalyptus species. Then we went for a skin prick test on 314 patients, found out 58% responsiveness, of which 36% were plus 1, 13% were plus 2, 8% were plus 3, and only 1% were plus 4. And rest were non-responsive. Then with the responsive patient, we further went for dot plot, found out that out of 96 patients, 27 sera showed prominent dot, rest were less prominent. We also did one-dimensional separation in 15% SDS page, found out 18 bands, of which 14 were very prominent, and we used a Princeton marker from 10 kDa to 250 kDa. We also found out the carbohydrate content was 5.5 mg per ml, total protein was 6.3 mg per ml, and lipid content was found to be near about 22% of the dry tissue weight. So with this, I would like to conclude that from the above findings, it could be said that eucalyptus is one of the potent source of aeroallergens and has a significant contribution in the seasonal respiratory allergenic diseases amongst the West Bengal populations. Thank you.
Thank you so much, Ms. Ghosh. So the session is open for interaction.
Okay.
Any questions for Ms. Ghosh from the participant?
One very simple question for you. What specific symptoms of this allergy?
Normally breathing problem or cough or nausea, etc. So bronchial asthma or allergy related problems.
How do you distinguish this specific problem for due to this eucalyptus pollen? It is not for other.
No, I am not distinguishing here. Actually, this is a primary basis because as we can see, besides eucalyptus, there are other aeroallergenic sources, different other pollen grains as well. So that's why we have to create a base for the entire study, that what are the prime contributors for these kind of allergy, and we found out that eucalyptus could also be a potent allergenic source here. So if we want to distinguish, then we have to go for further detailed examination actually.
Actually, so many allergens are there. At first, we have to distinguish this is specific for this type of allergen. Then we study the detail.
No, but what we do here is we first actually identify the different possible allergens, and then we went for SPT, dot blot, and ELISA and identify, because actually allergens are basically the protein, so they are cross-reactive to some extent. So we can opt for that.
Actually, what particular species of eucalyptus you have studied? There are different species.
No, I haven't mentioned the species here because it is not a species specific study, though the common eucalyptus vegetation here cultivated is Eucalyptus citroidora. So
Yes, now actually allergy disease is caused by a particular specific allergen, and that is the species specific also. So if you study the eucalyptus SPP, means the different species, that means that if you collect the pollen allergen,
so that is not species specific, but it is genus specific.
No.
Because
Allergy, it is the allergy sensitivity of the allergen molecule is very much species specific to human being. Eucalyptus citriodora may cause allergy among you, but it may not cause allergy to me. So it is the species specific. Aeroallergen is the species specific from taxonomic point of view. One thing. Another
species specific. Okay.
Yes, that is the species specific. So eucalyptus maculata may be allergen. It may be allergenic to you, but it may not be allergenic to me. Now, if you take the pollen samples from the different species mixed together, then the result may be some ambiguous, and that's why I have what particular species you have selected.
I have studied Eucalyptus citroidora.
Okay, citriodora, very good. Another one thing is that for the isolation of the protein from the pollen samples, it is very much difficult in eucalyptus species because the having of the essential oils, because oil is the very critical factor to isolate the protein. So what technique actually you have used for the isolation of the protein from eucalyptus pollen?
Free phenol method actually. So
Yes.
by phase separation technique, yes, first of all, we actually went out for different techniques and found out that only trisphenol gives the best result here.
Yes, right. Actually, I know because one of my friend, she has done a good bit of work in the eucalyptus pollen allergy from your same lab a few years back. Not a few years, many years back. Anyway, and that's why I'm asking. Thank you so much for the nice work and the nice presentation. Thank you so much.
Thank you, sir. Thank you so much.
Okay.
So, any more questions for the presenter? So then we'll move to our next presenter, Ms. Shampa Kundu from Palaeobotany and Palynology Lab, Department of Botany, Durgapur Vishwa University.
Yes, sir, I am present.
And I will request Ms. Ami J. Bhatt to please be prepared as you are the next one. So, Ms. Shampa Kundu, you can present your slide.
Okay. Sir, my slide is visible?
Yeah, it's visible and your voice is also clear. Go on.
Okay, sir. Thank you. First of all, good morning, my dear friends, my respected teachers and my colleagues. I am Shampa Kundu, research scholar from Sidhu Kanu Vishwa University under supervision of Dr. Mohsen Ali Khan. Today, I am going to present here first Cenozoic microfossil evidence of polypodiaceae from India and its biogeographic implication. fern, the most diverse and second largest non-flowering vascular cryptogams after Angiosperm and with 10,000 extant species. They were originated during late Paleozoic era but diversely distributed during Mesozoic and Cenozoic time. Among fern kingdom, Polypodiaceae, the largest leptosporingiate fern group placed under order Polypodiales. It is most diverse pantropical fern family. This family has rich fossil record from Cenozoic sediments in China, but unfortunately no fossil record has been reported from Cenozoic sediments of India. We report microsauroid fossil frond impression similar to modern microsaurum for first time from the late Miocene Shivalik sediments of Himachal Pradesh. Our main focus of this study to report a new fossil species of microsaurum from India, to review the fossil history of Polypodiaceae fern and its paleobiogeographical implication, and to trace its possible hypothetical migration route of Polypodiaceae fern. The age of the fossil almost 10.9 to 8.2 million years ago. We recovered our fossil sample by manual laceration by hammer and chisel, and after cleaning the fossil sample we took photographs by digital camera and kept it in our paleobotanical laboratory. This is our fossil locality, and this is our fossiliferous exposure, and this is our sedimentological litholog of middle Shivalik. This yellow part showing the sandstone area, and red part showing the clay stone or marstone type of sediment. And this is part and counterpart of our fossil sediment showing here. This is costal areoles including veinlets, and the veinlets ended with club-shaped tip hydrathodes. This is a line drawing of our fossil specimens showing the entire margin and prominent loop of secondary vein and tertiary and quaternary vein anastomizing from costal areoles. And this is our fossil specimen compared with a modern leaf of Microsaurum membranaceum. And we also detailed study of venation pattern of different comparable extant species of Polypodiaceae, and we took almost 20 morphological proxies to try to establish a dendrogram by PATH software, and showing here that Microsaurum sibalicum and Microsaurum membranaceum are almost similar, but some differences are also present here. This is hypothetical migratory route of Polypodiaceae. Before going to this topic, I want to show here detail fossil history of Polypodiaceae fern from during Cenozoic time in world. The maximum Paleocene fossil record from Europe, and maximum Neogene fossils were recorded from China. This fossil record unfold is intercontinental disjunct distribution theory, and we also suggest two possible migratory routes of Polypodiaceae fern. One is land bridge connection and another is long distance dispersal route. Might be the fossil Polypodiaceae fern migrated from Europe to North America by North American land bridge connection, and from North America to Asia, this family was migrated by Bering land bridge connection, and also there another alternative route that connected Europe and Asia due to closure of Targai Sea during Middle Miocene. And after coming Polypodiaceae fern in Asia, they were diversely distributed during mid Miocene to late Paleocene time, and migration of Polypodiaceae fern from China to India supports its into India dispersal distribution. And this fern also might be migrated from Asia to New Zealand, supports the long distance dispersal route. As the rise of the Himalaya, Eurasian and Indian plate form firmly sutured, and then migration of plant taxa from India to Southeast Asia mainland had occurred. And lastly, due to lack of fossil evidence, limits the understanding distribution and evolution of fern family in India, and so we need more megafossil record will able to shed light on the migration route of Polypodiaceae fern. And lastly, thank you, and I especially thanks to DST sir for funding us, and also thankful to my PI and co-PI. Thank you.
Thank you so much, Ms. Kundu. Now we'll request the participants to put forward their question. Any questions for Ms. Shampa Kundu?
Actually, I have a question.
Yes, yes, please.
Okay, Miss Kundu, that was a very beautiful presentation. Okay.
Thank you.
In your presentation, you have mentioned that the pteridophytes are originated from late Paleozoic era, right?
Pteridophyte originated during late Paleozoic era. Yes.
Yeah, but are you sure about that?
They
Because
originated after during Carboniferous time and diversely populated during Mesozoic and Cenozoic time.
yeah, but then have you heard about Cooksonia or Rhynia?
Yes.
Yeah, that
Mainly,
Cooksonia was
I was saying about Polypodiaceous fern.
then okay, but you have mentioned that
Yes,
pteridophytes
yes,
origin from
yes.
late.
Maybe.
Exactly,
Yes.
Silurian. It's originated from mid Silurian. That is the third
Okay.
period of Paleozoic era. So
Yes.
please rectify it.
That's maybe
Okay.
mistake. Okay.
Yeah. Thank you.
Thank you.
Any other question for the presenter?
Yes, it is a very good bit of work, a very good and compact work.
Okay.
One thing, and thank you for the good for doing your good work, and presentation is also very nice. One thing is that did you observe any differences between the extinct Polypodiaceae and the
Yes. Obviously, I observe.
and particularly with reference to the Venetian pattern in the evolution of the Venetian or something else? Is there any differences?
Yes. Let me show. This screen is visible.
Yes.
Yes, visible.
Okay. Maybe the picture might be blur, but I try to see.
Just Venetian, regarding the Venetian.
Okay. I try to see. If we see Microsotum membranaceum, the costal areoles arrangement is almost regular shape. But in our fossil specimen, costal areoles arrangement in irregular shape. And if we see the secondary vein divergence of secondary vein angle is less acute, whereas our secondary divergence angle more acute, and some differences also present here. Microsotum membranaceum marginal is quite wavy than our fossil specimen. Present
Yes.
Microsotum margins.
Yes, I see. So here, what type of theory might be applicable? Actually, from theoretical point of view, we know the different theories are there, like the innocent theory or weaving theory, like that. So here, the what type of theory might be applicable for this type of the change?
Sir, we are going for this study, and we find for more fossil collection, and after that we try to establish a relation between this study. From one fossil collection we
Very
cannot
good.
say it.
It is very actually Palaeobotany is very interesting work, and you have done a very good work. Okay,
Thank you, sir.
so much.
Thank you, sir. Thank you very much.
Okay.
Thank you. And then any other questions for the participant? Okay. So we'll move to our next presenter, Ms. Ami Bhatt from Talod, Gujarat.
Hello, am I
It's a
audible?
plant cell science college. Yes, you are absolutely wonderful. And I will notify Ms. Rituparna Shaha to please get ready for her presentation, as she is the next one. So, Ms. Ami Bhatt, you can present your slide. Thank you.
Is my screen is visible?
Till now, no. Should we share from our side? Okay, it's coming. It's coming.
Now it is.
Good morning, everyone. I myself, Ami Bhatt. I am from Gujarat. I am pursuing my PhD on some anatomical work of some weeds from Banaskantha, and here I am presenting some patch of my work. Uh, my poster is on comparative anatomical study on trichome and stomata of 5 asteraceae weeds plants found in the crop fields of Banaskantha district, Gujarat. Banaskantha district is located in the northeast of Gujarat, and it has a semi-arid climate, and the major part of the soil are sandy in nature, and bajra, maize, tobacco, castor and jawar are the major crops of the district. The Asteraceae family has an annual, biennial or perennial herbaceous plant, and there are some shrub and trees are also present. Plants of Asteraceae family are economically important as a weed, ornaments, medicinal or some as a green vegetables. I'm presenting your study on a stomata and trichome. The stomata are aperture in epidermis. Stomata are bounded by two guard cells, and the different types of stomata are classified on the basis of the number of surrounding secondary cells. In these 5 species, I observed mainly 3 types of stomata. First one is anemocytic, and second is anisocytic, and third one is actinocytic. And then the main function of the stomata is to control water loss by transpiration, and they allows the gas exchange for the plant. And move to the trichomes. The trichomes are highly variable in structure and function, and they are mainly classified into 2 types. First one is glandular, and the second one is non-glandular. And mainly I observed unicellular, glandular, simple or multicellular trichome in these 5 species. And in the Borndonia scenario, I observed a glandular or T-shaped trichome. And Eclipta prostata has this unicellular glandular trichome. It has a tip on the trichome. You can see in this image. And then main function of the trichome is a reduce or transpiration rate, and work as a water storage organ also. And granular trichome are trichome secreting a poisonous substance protect the plant from the animal attacks, and also nectar secreting trichomes helps in a pollination to attract insects, and some digestive trichomes are helps in capture and digestion of insects. Uh, this is a small work from my PhD work. I am doing the work on Wool district. I covered 2 talukas up to this, and I mainly observed 7 species of Asteraceae family, and the Asteraceae family is abundant till now. And there are some further 2 plants, Urticaria and Sonkus were present there, but the anatomical work still not done, so I am not presenting there here. Thank you.
Thank you so much, Ms. Bhatt. So the poster is open for interaction. So any question for Ms. Bhatt?
Yeah, I have a question. What could be the application of this kind of work? You have studied the different types of stomata and different types of
Yes.
trichomes of this family.
Actually, I'm studying totally anatomy of whole plant, but here I'm presenting some little
Studying
work from
anatomy is not an application. What could be the application of this kind of study? This kind of
Actually, most of the
morphological study?
weeds have some medicinal properties too, and right now the pharmacognostic study are in trend. So the anatomy is the basics for the
Actually, madam is asking you that whether such type of study is applicable for the forensic botany or archaeobotany
Yes,
or
Yes, yes.
drug
Any type
analysis,
of field of botany.
any
Yes.
applied aspects. Actually, madam is asking for any crude drug analysis or the organoleptic
yes,
study
sir.
or the
For
archaeobotanical study or the application in the forensic botany. Like this, madam is asking. So you try.
after taxonomy or a morphological study, the anatomy is the only key that you can find the exact species or exact identification of species by the anatomy. So this is the base of the work. I can say that.
Do you think that anatomy is the final option for identification of a species?
No, not after that the microanatomy or micro molecular biology also present there. But
What kind of
after
molecular analysis for species identification is there?
I don't know. So,
I have one question, sir. You have studied stomata and trichome. Among these two, which one is taxonomically important for the genus identification of this particular five genera?
sorry, ma'am, I can't hear you properly.
You have studied trichome and stomatal structure, okay? Among these, which character is important for generic delimitation? These are the different characters you studied, what are different types of stomata, different types of trichomes. But taxonomic point of view, what type of stomata or the trichome is important for generic delimitation?
No, ma'am, I don't know.
Any other question from Ms. Bhatt?
Yes, good morning. I have a small question. It's actually a commendable work that you have started with the anatomical work. Before we go on for further investigations, this has to be done to determine the species. Fantastic. Now, you have taken only two of the aspects anatomically. Why did you not consider the vascular bundles and other specific features also? Number one question.
Actually, ma'am, I'm studying a whole anatomy of a whole plant, but in this poster, I only mentioned this.
Okay. Now, in that, two of the species that you have mentioned about Parthenium and Tridax, they do have similar characteristics with the two aspects that you have taken, that is stomata and trichome. Are you planning for further differentiation anatomically itself?
Yes.
So what is that you're planning to do?
I'm going to calculate stomatal index for this.
Okay, very good. Okay, nice. Thank you. What other further works you're planning to do?
I'm creating data on base for this area.
Okay, because you have taken the weed characteristics and you say that you're also doing the complete anatomical study on the weeds. So what is the target? What are you targeting? What is your plan ultimate? Why is this work being taken up ultimately?
Actually, we show weed as unnecessary plants, but weeds has then own characteristic and own uses or own medicinal values there. And मतलब उनकी कोई ऐसे बेसिक वर्क और सबकी एनाटॉमी में मिलती नहीं है, तो मतलब मैं वो करना चाहती हूँ. और बनासकांठा डिस्ट्रिक्ट हैज सम डेजर्ट एरिया आल्सो, so some habitat changes can I can.
Okay, I have a small suggestion for you. If you go for further phytochemical studies, it will be very useful in your findings. You will be able to find a lot of differences and new ones. So just a small suggestion. All the best to you.
Thank you so much. Sure.
Thank you so much, Ms. Bhatt. So we will move to our next presenter, Rituparna Saha from Molecular and Applied Mycology and Plant Pathology Laboratory, Department of Botany, University of Calcutta. Saha,
Yes, sir.
can you please present your slide?
Yes. Is it visible?
Yeah, visible, absolutely visible, and your voice is also clear. You can go on.
Today I am going to present the work First Record of Tophagus catenensis from India. The family Ganodermatidae belonging to the order Polyphorales currently have 11 genera including Tomophagus. The member of Ganodermatidae mainly characterized by unique double-walled truncate basidiospores. Scientist Muriel segregated a peculiar species Ganoderma colossus from the genus Ganoderma based on its unusual thick and pale context, which becomes soft and light after drying, and created a new genus Tomophagus to place the species as Tomophagus colossus as a type specimen. A second species of Tomophagus catenensis, was discovered for the first time from Vietnam in 2012. Later, it was reported from Pakistan in 2022. Till date, these two species are reported under this genus with restricted distribution. A unique species was collected from Kollani, Nodia district of West Bengal by our research group, and thorough macro and microscopic characterizations and ITS-based phylogenetic analysis revealed that this unique species is Tomophagus catanensis. The major observation of my work is the major objectives of my work is at first we collect the specimen from different parts of West Bengal, then we identify it is based on morphological and molecular protocol. This is the field picture of our collected specimens. Some important macroscopic characters are here the basidiocarp is solitary, perennial, bulbous, spongy, easy to crack and light in weight after drying. Pileus upper surface azonate, glabrous and lacquate at first, later become dusty and brownish orange. Context greyish orange, and the pores are angular 2 to 3 per mm and tubes are not stratified. And here some microscopic pictures are this specimen has cutis layer. This cutis layer is composed of closely packed clavate cells. These clavate cells are looks like this A to B, and this is the basidium with 4 stegmata, and the specimen has diametric type of hyphal system. The generative hyphae is clamped and the skeletal hyphae is thick walled with narrow lumen. This specimen has chlamydospore in the context and the trauma region, and the chlamydospore is intercalary in position, and the specimen has the basidiospore which are typically truncate, bitunicate, ellipsoid in shape. Here in the n number of picture, exospore is hyaline and the endospore is brown with compact reticulation. After thorough macro and microscopy, we made a phylogenetic tree applying XML and Mr. Bias using the best fit model GTR I plus G model. Here we took 2 species of Trematis as an outgroup, and both the ITS sequences of Tomophagus colossus and Tomophagus catanensis along with all other Ganoderma, Sanguinoderma and Amioderma species was selected to make this phylogenetic tree. Here this tree clearly shows that all the Tomophagus catanensis species clustered in a same clade with 100% bootstrap support value. Here we took the type specimen of Vietnam, type sequences of Vietnam, and the CUHAM 504 is our newly generated sequence, and these are clustered together with strong support value. So we can say that our specimen is Tomophagus catanensis. And extensive literature survey revealed that there is no previous report of Tomophagus catanensis from our country. Hence, the present finding confirms the first report of Tomophagus catanensis from India with a new addition to the macrofungal flora of India. Thank you.
Thank you so much, Ms. Shah. Now the post will open for interaction. Any question from any participant for the presenter? Any question for the presenter from any participant?
Yes, actually, it is a good one. I just like to know what are the basic taxonomic procedure for documentation of any new record.
At first, we have to do macroscopy and microscopy. Macroscopy, we have to note some field data during the field collections, such as pileus, upper surface color, texture, presence or absence of hair, or presence or absence of stipe and their attachment with the substratum. And then we have to do the microscopic characters. Some typical characters are such as basidiospore shape, color, we have to check the chemical reactions that the spore or the hyphae color change or not change, or dextrinoid or ardextrinoid or amyloid in presence of Melzer reagent. And, we have to check all these things. Then
Yes.
if we see that the our specimen is not matches with other specimens of our in a key under a genus or a family, then we will do the molecular analysis.
No, before that, the regarding the basic collection procedure or something else, there are some maintenance of some type materials, or regarding the keeping of your voucher specimen. What you will do?
Yes.
Actually,
Yeah.
for further work, you will do the molecular analysis and for
Yeah.
establishing
Yes.
your report. But before the going to the molecular work, you have to do some other work also for the documentation purpose. That means the voucher specimen, ready references for the voucher specimens, or
Yes.
any
Yes.
type
We
is there or not. That you have to consult that thing.
Yes,
Yes.
we at first we collect our specimen, and we to preserve a specimen for a long time, we have to dry it in a 60-watt bulb
Yes.
to absorb the moisture, to remove the moisture. Then we after removing the moisture, the dry sample, we have to preserve it in a silica gel in a packet, where we put a silica gel some amount, and we preserved it within them, so that if moisture comes, then the silica gel absorbs the moisture, so that we can keep our specimen for a long time, at least 10 to 15 years. And we preserve our sample in our lab in Calcutta University Herbarium in such a way, type material, new record, all of the samples.
Yes. That's why. Okay, thank you so much. It's very good work. Thank you.
Thank you, sir.
Any other question for Rituparna from any participant? Okay, thank you, Rituparna.
Thank
So
you, sir.
we will move to our next participant. Last but not the least, this is the last one. Mrs. Motushi Shen. She's a faculty member of our department and also an member of this webinar organizing committee from Department of Botany, Durgapur Convent College. Just for information, this poster will not be a part of competition. She is here to present her work, although the poster is open for interaction and discussion. So, Mrs. Motushi Shen, are you ready with your poster?
Yes, Shandipan, I'm ready.
Yeah, you can share your slide, please.
Thank you. Is it visible?
Yes, it is visible.
Visible.
Okay, thank you. Good morning, everyone. Respected principal sir, my dear colleagues, present delegates, and my students. The topic of my poster, today's poster, is comparative micromorphometric studies of leaf epidermal cells of few Fabaceae members. So, first, before I enter in this poster, I want to discuss about the background. The objective of my study is the study of the cell division and elongation pattern in the leaf. So, why? Any organism as well as the organs development developed by cell division or cell elongation. These are two procedures. Our main interest is to study the growth pattern in different plants in nature. For this, we select leaf epidermal cells. Why? In this case, leaflets. Leaflets are bilateral outgrowths, and growth of leaf corresponds with the growth of epidermis. So, leaf epidermis shows different types of outgrowths and special cells which are much more sensitive to environmental changes. Their presence, their size, shape, appearance, these are different in different environmental, they are sensitive in these environmental changes. So, we target the pavement cells. Now, for that, we just select any plant from this family in our locality and select the leaflets, two types of leaflets. One is the smallest one, another one is the largest one. Now, our interest is to study the cell size in the smallest one and the largest one. And the cell elongation is a procedure which is found in the epidermis. So, we collect small 2 or 3 successive leaflets and the largest one by the help of the scale. Then we study the adaxial surface, because these outgrowths, stomata, trichomes, others are much more in the abaxial surface. So, we prefer to study the adaxial surface for our pavement cells. And then we photograph, we see their structures with the help of the microscope. And 1 minute. And then measure the cell sizes from each of these with the help of image J. And we measure the apical one, middle one, and the basal one, because there are differences in this. And also the leaf development shows differential growth pattern in the apex and the base. In some plants, the apex shows meristematic activity later in their developmental time, whereas the reverse found in some plants, in some leaves, that is in the basal parts, shows the meristematic development for long time, and in some cases throughout the leaf it's found. So we take each of these measurements depending on the average of upper, middle, and lower. Among these, there are Cassia fistulas, Sena spectabilis, Cajanus cajas, Delonix regia, and this is the list which is depending mainly on their availability. Now, when we take this large epidermal cell area and the small epidermal cell area, we can see that there is a distinct cell size differences, cell area size differences in this group of plant, where in Cassia fistula it is only 347 micrometer squares in the diameter area is, whereas in Lathyrus it is 2584, and the in the small leaves, the epidermal cell area is also vary, but it is not very as it's vary in the large leaflets. Now, the cell expansion ratios are calculated by dividing these large epidermal cell area divided by the small epidermal cell area to represent how much it expands. And this is the small cell area from the small one, and we found this. Now I am coming to our discussion and the findings. We found on this, when we arrange these accordingly, these are the rates are the tree members. These are the shrub members, and these are the herbaceous members, and they shows a distinct pattern. That means the tree members or perennial members, in case of this, the cell elongation is lesser, and in the herbaceous, cell elongation is much more than the tree members. So from here we can say that this is a preliminary work. We can say that the for cell division we need much more energy than the cell elongation. So the herbaceous, which have a small lifespan, they have to complete their life cycle within that time with limited resources. So they are rely much more on the cell elongation and use their energy for reproduction, whereas these tree members maybe have such resources which they can use, or they are least bother about this. This is our hypothesis. This is the latter works are going on, and I want to acknowledge our principal sir, our head of the department, Dr. Ashok Bhattacharya, and my departmental colleagues, the Dr. Shikha Datta, HOD of Department of Botany, Bardhaman University, and all of who helped me in this work. And these are the references. Thank you.
Thank you so much, Mrs. Sen. Now the poster is open for discussion and interaction. Any question for the participant?
Yes. I think now that we are very lucky enough in today's morning that we have got our one of the colleague, Motushi Sen madam. Actually, the webinar is nothing but the digital platform for exchange of the views and ideas among the different scientists. So here the platform, it has been organized and going on by us. But in that sense, we need also to exchange the views and ideas from our behalf. So I am very much thankful, and I thank Motushi madam for presenting the nice work only in purpose of the exchange of views and ideas of our department among the other participants throughout our country and the other departments. Many other departmental, many other institutes of our country, they have participated in the poster competition. But thing is that without any competition nature, competitive mentality, you have shown your credentiality among the participants to explore the views and ideas of our department among the other participants without any competition. That's why we are very much thankful to you for your nice work and the nice presentation. And your work is very nice in that sense that not only it will emphasize on the analysis of the taximetrics or the numerical taxonomy, but it also correlates some ideas regarding the energy conservation process with respect to the reproductive mechanisms. So that type of the work, the cell elongation and cell division study and its correlation with the taximetrics as well as the reproduction and the energy conservation rules, all these things are very greatly correlated, and I think it is the very new one. And wish you all the best and in your future academic pursuits, and wish you best of luck. And we are very much thankful to you, madam, for presentation of your nice work in this platform.
Thank you, sir. Thank you, sir. But it is not my only work. It is my work, and the presentation is only possible for my colleagues, including HOD. And I also want to mention the name of the Anish Bhattacharya. He is the one who always said তুমি দিয়ে দাও দিদি, একটা পোস্টার দাও, আমাদের থেকে একটা যাক, লাইক দিস। সো থ্যাংক ইউ all. Any other
Any other comments?
Excellent presentation, Didi.
thank you bhai.
So then we can, I'm calling again the name of Mr. Parth Patel from Department of Botany, University School of Science, Gujarat University. Are you present here, Mr. Patel? Or any other member from co-authors of the poster? Any other? Miss Jyoti, Miss Nilam, Mr. Dipesh, Miss Suhani Parekh? I guess not, they are not present here. So then we'll move to our next session, that is the plenary session and the plenary speech. But before entering the session, actually, I want to share a very sad news to all of the presenter and all of our participants that we just came to know last night that our eminent speaker, Dr. Sandeep Kumar Behera, scientist, NBRI, will not be able to join us in the webinar due to a sudden death in his family. So his lecture is scheduled from 1:25 p.m. today, but we have to make a little change in that. We spoke to him and requested him if he can manage to give a talk, as we don't want to miss his lecture. So he agreed to deliver his lecture on Sunday, 26th of June 2022. So we will reschedule his lecture, the prize distribution, and the valedictory session on the Sunday, starting from 10 a.m. in the morning. It's our earnest and humble request to all the participants to look into the situation and to present a few more hours with us actually on Sunday, and to make the event a successful one. We are extremely sorry for the inconvenience. Fresh schedule will be sent to you shortly. Thank you. So now we will move to our plenary session. Mr. Anish Bhattacharya, can you please continue with the session?
Yeah. Am I audible?
Yeah, you're audible.
Okay. Thank you so much, Dr. Ray, for giving me the opportunity to introduce our first eminent speaker of today's session. I'm going to introduce that person to you who is continuously creating numerous deserving botany warriors for many years. It is my privilege that I am one of them who got the chance to be her student and witnessed her excellent way of teaching, not only inside the classroom, but also in the field. I do not know whether madam can recognize me or not, as I have changed a lot from my college days. Okay, so leave it. So please allow me to welcome Dr. Ashalata D. Rosario. Dr. D. Rosario is a retired associate professor from Narasingha Datta College, Howrah. She did her MSc specializing in Palaeobotany and Palynology, and PhD from University of Calcutta as an University Grand Commission Research Scholar. She is the recipient of CAS TWOWS Visiting Scholar Award in Beijing, China, in the years 2006, 2009, 2011. She has attended a number of national and international conferences and seminars, visited places like Germany, China, Japan. She has experience of more than 31 years of teaching in the undergraduate classes and has been teaching in the postgraduate classes of different universities and colleges in West Bengal since 1994 till date. She has a number of publications in both national and international journals. She is the member of International Organization of Palaeobotany Palynology, Botanical Society of Bengal, of which she is presently the secretary. So it is going to be a great opportunity mainly for MSc and BSc students to acquire lots of knowledge, and for me too to listen to her as a student again. So I would like to request Dr. Ashalata D. Rosario ma'am to deliver her speech. Madam, please.
Thank you so much. I hope you can hear. I am audible?
Yes, ma'am, you are completely audible.
Okay. Thank you so much, Anish. I do remember you very well. And good morning to all of you.
respected Principal Sir, Dr. Devnath Pali, Joint Conveners, Dr. Ashok Bhattacharya, Dr. Prashant Saha. It's my honor and privilege to present before you in this seminar, in this webinar rather. When Dr. Prashantu approached me to say something about gymnosperms and paleobotany, I just could not refuse in spite of all my other schedules. I love saying something to the students, and so I did not miss this chance. These are two vast areas, as all of you know, and I mainly focused on taking the students as I was told, with the undergraduate students through the world of gymnosperms and through the world of paleobotany. Generally, students do not like these topics so much, but I would tell you that these are very interesting groups, very important groups, and one need to have a knowledge about it. So let's go with the slides. I'll share my screen. Just hold on. Okay. So I hope you can see. Okay. Now, gymnosperms, the naked seeded plants. Now, as you all know that the seed bearing group, we have the gymnosperms and the angiosperms. Gymnosperms, where the seeds are naked. And what does this naked seeded plants mean? They are flowerless plants that produce seeds which are not enclosed by or not enclosed within the ovaries or the fruits. Unlike angiosperms, where the seeds are enclosed within the fruit. The term gymnosperm was coined by Theophrastus. The study of gymnosperms is called gymnospermology, and the word comprises of two Greek words, gymnos, which means naked, and sperma means seed. So that is gymnosperms. In general, there are four distinct divisions among the gymnosperms. I am mainly focusing on the extant gymnosperms, and generally in all the books you will find such groupings are there in a broad sense. There are many systems of classification. I am not going into the details of the classification, but almost all the classifications, these groups are there. The psychodophytes, the coniferophytes or the psychodophyta, coniferophyta, gingophyta, and gnetophyta. And with about 88 genera and more than 1,000 species, they are distributed throughout the world, both in the temperate and in the high altitude regions of the world. Now, as you can see over here, that in gymnosperms, in angiosperms, sorry, we have the gynoecium, the stigma, style, and the ovary. The ovules are within the ovary, and that makes it closed. In gymnosperms, there is no carpel as such, so the ovules are exposed. They are borne on modified or unmodified leaf structures, which are known as the sporophylls. So the megastrophyll. Coming on to group wise, let us go through the psychophytes. The psychophytes, they are woody plants. Usually they are trees, generally unbranched, resembling a palm tree with a cylindrical stem and a crown of pinnate compound leaves. The group is variously divided. Some consider that there are 3 families, some 1 family, whereas some say that there is a single family, Psychadaceae. At present, we are all more or less familiar with cycas, the common psychophyte that we see around us. Many of us have in our homes or gardens, almost everywhere you can see it. But there are many others. At present, there are 11 extant genera. Dione, Ceratosamia, Zamia, Tigua. Tigua is the latest addition. Microcycus, which all belong to the Western Hemisphere, and the remaining 6, that is Cycas, Macrozamia, Lepidozamia, Cephalotaxus, Stanzaria, Bowenia, occur in the Eastern Hemisphere. Cycas is commonly found in the tropical and subtropical regions of the world. So you can have a look at the Cycas plant. They are from various places which I have collected, and most of these photographs are my own taken photographs. The first one you can see, this is from Scottish Church College. They have a lot of these cycas plants, so I took a snap from it. This is in another place. Generally, I said they are unbranched, but in many cases, if the tip is injured or broken, sometimes we see branching arising. So you get a branch structure. So it almost appears like a palm tree. The leaves are very characteristic. They are pinnate compound leaves at the apex, and the leaves show sarcinate vernation, a characteristic feature of the fern. So in cycads, we will find a lot of these fern characters. Then they have the reproductive structures, the male and the female cones. Cycas is dioecious. Male and female plants are separate. Males, there are cones, well-developed cones. In general, gymnosperms have well-developed cones. I have not gone through the characters of gymnosperms because I said that it will be better to say along with the different groups. Students will understand better when I show the photographs. So in the gymnosperms, we have all male and female cones are there, but with the exception of Cycas female, there is no female cone as such. We find over here that the megasporophylls are there compactly arranged. When they mature, they will become loose, and we get a structure like this. So this is the megasporophyll, and the megasporophyll bearing the ovules on the 2 lateral side. And very interestingly, one has matured. This is collected from Bribon College. They also have a lot of these cycas plants, both male and female. So this is one which has become matured. And when it matures, it becomes bright orange red in color, very attractive to the birds. And when they are young, they are green in color. So this is in one species. This is in another species, Cycas revoluta. So I just put 2 of them to show you how different they are. And they are very characteristic of species. So you can identify these species based on the structure of the megasporophyll. They are very, very characteristic. These are the male cones. This is again taken from a Scottish Church College, where there was a male cone developing. It grew much bigger, and then I told them to cut it off and preserve it, because otherwise it will all fall. This is from Bribon College. The other day I just made them cut this. I was there to make it ensure that they are well preserved. This is from another species. So you see how the male cones are there. Each of them is a microsporophyll. And you see how big they can be. This is just a week before, maybe 10 days from Bribon. And you can see the pollen grains, the yellow dusty powder, the pollen grains which are there. So again, a closer view. So on the abaxial surface, you get the microsporangia which are aggregated in clusters forming a sorus, another fern character. So a single microsporophyll, this is a preserved one, single microsporophyll, and you get the sorus on the abaxial surface. This is another member of the psychotophyte, very common. This is Zamia. Cycas and Zamia are very commonly found here. So Zamia, you see the leaves, they are quite large. They are pinnate, no doubt, but quite large, unlike cycads which are long, linear. So you see many of these cycas, Zamia plants over here. And you see the cones. These are the female cones. I will show you a more better picture. So these are the female cones. You see so many of them are growing, coming up. So many more Zamia female cones are there. The female cone may be so big, and when we make a section, a cross section of it, you see that how the ovules are arranged. So we get the ovules which are pendant. In cycas, I showed you the ovules are borne on the lateral side. Over here, they are pendant in nature, borne on the lower surface of the megasporophyll. These are the ovules which are there. And this is a sectional view of it. Cycads are important also in many ways. As students have read in books, that cycas, the stem, there is a lot of starch produced from the stem. Even in the seeds, which are edible, as I told you, they have profuse amount of starch. And from starch, which is commonly known as sago, lot of edible items are prepared from it. Cycads, they have cycacin, the active principle. Active principle is cycacin, which is medicinally important. And we see that the young shoots, they are eaten as vegetables. Besides that, the leaves are used for thatching roofs, etc. So more or less, to give you an idea how psychotophytes look like, what are their reproductive structures. So the psychotophytes, including the cycas, Zamia, there are many other genera. As I told you, there are 11 extant genera. Coming on to the conifers, the most important group among the gymnosperms and the largest group, and very important in many ways. In many ways, they are very, very important. We find that the conifers, they are the world's largest timber yielding plants. The paper industry depends upon it. We get resin, we get copal, we get sandarak. Then our common laboratory mounting media, Canada balsam, is obtained from the conifers, Abies balsamea. And even they are very important as drug yielding plants, as well as they also are, some of them are important as food. We will go on with these characters as we go along with the different families. Now, the conifers, in contrast with the psychotophytes, they are tall trees growing in the temperate regions and high altitudes of the tropics. They are woody perennial plants, profusely branched, bearing leaves of different types, maybe needle-like or maybe more expanded. And within this coniferophyte group, we have the world's tallest trees, the massive trees, the smallest gymnosperms, all are within this group. And they date back to the Carboniferous. But presently, we find there are some extant members. They generally have two types of branches, the long shoot and the dwarf shoot. Long shoot of unlimited growth, dwarf shoot of limited growth. The leaves are also dimorphic. They have scale leaves and the foliage leaves. The roots are mycorrhizal. They have pycnoxylic type of wood in contrast to psychodophytes which have manoxylic type of wood. And being pycnoxylic, the wood is economically important. The reproductive structures are in the form of unisexual cones, male and female. The females are compound in nature. We will see how they are compound and complex in nature. The males comparatively are simple. The pollen grains may be winged or non-winged, and the embryo may be dicot or polycotyledonous type. Conifers generally divided into 7 families: Pinaceae, Taxodiaceae, Cupressaceae, Podocarpaceae, Orocariaceae, Cephalotaxaceae, and Taxaceae. We will now have a glimpse into the different members of these families, some of the representative members of the families. So, Pinus, very well known to all. I don't know in this pandemic if students have been able to go for excursions, but generally when we go for the excursion, we see Pinus. These are the different species, different types of cones which are there, the female cones. So you can see that how there is variation in the cones depending upon the species. This is the male cone. The male cone, they arise in clusters. They arise in clusters, and each cluster, they are not very large, small, about 3 to 4 centimeter maybe, consisting of profuse number of microsporophylls, each microsporophyll bearing 2 pendant microsporangia, and they contain millions and billions of these pollen grains which are winged. And these female cones also, depending upon the species, you can identify the species depending upon this cone structure. So this is Pinus armandi. Just to give you an example, this is another species of Pinus. So this is from Pinus wallichiana. They have quite elongated cones. If you go to the Himachal Pradesh, you will get these type of Pinus plants growing there with these type of cones. This is another species, Pinus gerardiana, Roxburghi. You have, this is Pinus gerardiana, this is Pinus roxburghi. Then you see the sporophylls, the megasporophyll. Actually, why this is compound? Because this axis, they consist of, when we see in the young cones, such as the slides that we see in the classes, we find that there is the brachyscale and the ovuliferous scale. The brachyschale is there, and at the axil of the brachyschale is the ovuliferous scale. So they are fused. In case of Pinus, they are completely fused. We will see in the other members, they are partly fused. So, and you get the 2 ovules bone over here. You can see the impression where the ovules were there. So this is the dorsal view, this is the ventral view where the ovules are there, and again more clearly where the seeds were there which have matured, and the seeds are characteristic. They have this wing. So the wing seeds, they are important features of Pinus. And this is another, this is the mature seed. Seeds we can see over here. This is one species seeds over here. The seeds are attached with the wing, which helps in dispersal. And these seeds, they are generally known as, or commonly known as, chilgoza, obtained from Pinus gerardiana, chilgoza pine. They are eaten, roasted and eaten, so as nuts, So I had got a present of the dry fruits, and I came across this one segment of that box, rather, with these seeds, with these roasted seeds. So I kept them, and to show the students. I keep showing them because you get a rare chance to see them. So this is Pinus gerardiana chilgoza pine, and this is the winged pollen. We come to the next genus, that is Cedrus. I will go fast with these slides because a lot of things are there to cover. So Cedrus, again we find the leaves are needle-like. Pinus, as you know, that they have needle leaves, but not all conifers have needle leaves. So these are the female cones. They are still immature. So these were taken when I used to visit China. So a lot of these trees were there, a lot of gymnosperms are there, and most of the pictures are from there only. This is the male cone, and this is the female cone. So, the male cone, a female cone, the male cone again consists of the microsporophylls, and the female cone, as you see, that these are the megasporophyll. This is the seeds which are there. Again, we have the wing seeds. And these cones, in case of Pinus also, in Cedrus, they are quite woody in nature, and they are used for decorative items. When you go to the hills, you can see that they sell a lot of these decorative items made of these Pinus cone. When you go to New Market, you will find a lot of these cones are there. Cedrus, they make dry flowers. So they are used for decorative items also. Then this is another genus, that is Larix. Larix. They are tall trees, and you see the cones over here. The cones over here, Larix. So the female cones, these are the female cones. Comparatively, they are small, comparatively small. Another genus, Piscia. Another genus, Piscia, all within the Pinaceae family. We come to the next family, that is your Taxodiaceae. Taxodiaceae. Taxodiaceae, we come to Cryptomeria. When we go to the eastern Himalaya, so as we ascend up from NGP, climate becomes cooler. We are ascending, we see these tall trees around, these are all Cryptomeria. Cryptomeria japonica is the common species. So the male cones which are in clusters, and these are the female cones. Comparatively smaller, but woody in nature. And in these cases, you see some spine-like structures covering the entire surface. They are actually the tips of the ovuliferous scale, which have not completely fused as we get in Pinus. Then this is Metasequia, very, very important, native of China. So in that institute where I used to visit, Beijing Institute of Botany, Chinese Academy of Science, they have a huge garden. It's much bigger than our botanical garden in the institute. Although right across there was a botanical garden, but so you see that the two sides, they are all planted with this Metasequia trees. Metasequia glyptostroboides, the only species commonly known as the red dawnwood. Red dawnwood. A closer view of the leaves, which somehow I had brought it to show the students again in the classes, the leaves, how they look like. So these are the plants, the red dawnwood, known as the living fossil. The interesting point is that this plant was found as a fossil first and then the living plant. And we get a plant in the Lloyd Botanical Garden also. There is a Metasequoia plant in the Lloyd Botanical Garden. Another important plant. So this family is very important. Sequoia sempervirens, known as the Californian or the coast redwood, which is confined to California. And these pictures, actually, this is my friend's son who stays in California. And all these pictures, when they used to send to me that they have visited there, I would keep them so that someday or the other I would get a chance to show these students. So they are all taken by them from there. So you can imagine how tall it is. The characteristic feature is that you see this almost 3/4, more than half, they have no branches. The branches are confined over here. And these are the reproductive structures. These are the reproductive structures. This is the male and this is the female. So, Sequoia sempervirens. Another important plant, Sequoia dendron giganteum. Sequoia dendron giganteum. Again, these photographs were taken by my friends who had been there. You will get these to see in the books also. So, Sequoia dendron giganteum, the massive tree, or the big tree as we call them. And so huge is the diameter that even when the central part is removed, you see a car easily passing through it. People often go there and they take photographs. My friend, Dr. Shupriti Daktu, who was in Noihati College, the morning section, Noihati College for Women, I think is the name that. Her two sons who are working in Apple, staying in California, they had gone there and sent these pictures. So, Sequoia dendron giganteum. Another important plant, Taxodium. Taxodium distichum. And you can imagine from where this plant is. From a Acharya Jagadish Chandra Bose Indian Botanic Garden. Many do not know about it. There is Taxodium, so I have kept this label also. So Taxodium plant growing around a pond. Only Gymnosperm which grows in the marshy areas, halophytic region. And thus they produce these pneumatophores. So around the tree, you find these pneumatophores. I have enlarged it to show you how these pneumatophores look like. It's in our Indian Botanic Garden. So, Taxodium distichum. We move on to the next family, that is Cupressaceae. Cupressaceae is characterized by plants which have scale leaves. Scale leaves. So, this is Cupressus, full of these female cones, still immature. This is slightly enlarged view, and see, this is a matured one. And these tips, they are nothing but the remnants of the fusion of the bracteal and the ovaliferous scale. And inside it, you find the seeds. So when they mature, the seeds are dispersed and you get it. So this is a Cupressus. Another allied genus is Juniperus. Juniperus. So this is how the plant looks like. We have in our gardens also, many people plant it, have in their pots or in their gardens, Juniperus. Another plant, many people say this is Thusa, but Thusa does not grow over here. I had seen Thusa in China, Japan, Germany also. But Thusa does not grow over here. They look like Thusa. The leaf, when you touch them, they are flat. They are flat. You can keep inside your in the books, within the pages. But juniperus, they have these spiny leaves all around. So this is thusa, which looks like thusa, but actually this is biota. There are certain differences between thusa and biota. They look very similar, but careful investigation, which I make the students work out in their MSc classes, and they can see the differences. So this is the male cone. They are very young still, not yet mature. They slightly become larger. The characteristic feature is the leaves. They are arranged in opposite decussate manner. And these leaves, you see central region, they have a groove. Either they have a groove or a gland. Either they are glandular or they have a groove. In thusa, there is a gland. In biota, there is a groove. Opposite decussate manner, it is arranged. We come to the female cones. Female cones. And we see that these are the female cones. And it is some few days back only I just took out these parts, the megasporophylls. So you see the pointed structure, again the remnant of the ovuliferous scale, which had already fused with the bracteal and the ovuliferous scale. And inside you see the seeds are there. These are still immature. When they mature, they become larger and very hard, very hard. And again, on the other side, you see the grooves are there. And in biota, when you make a section, the outline is triangular, typically triangular, when they mature. In thusa, it will be oval structure. So that is how we differentiate thusa and biota. Besides that, there are many other differences. I just mentioned the differences that we see from the figures. We come to the next family, that is Podocarpaceae. The most important genus is Podocarpus. 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 Again, Podocarpus, they are somewhat larger trees. We get in a botanical garden also. There are many Podocarpus plants growing there. But these I collected the photographs from China and Japan. So Podocarpus, you see, the leaves, they are quite flattened, unlike the spiny leaves of the other members that we saw. And these are the female cones. These are the female cones in large view. The very characteristic, these are the preserved ones. I just showed you, just put it there to show how this is a receptacle. At the base, there is a receptacle. This is the ovule. And this ovule is again covered by a structure which we call it as the epimetheum, a fleshy structure. But the characteristic feature is this receptacle, which is brightly colored. As you can see from the photograph, it is very brightly colored, which is the receptacle. This is the characteristic feature of Podocarpus. Coming on to Orocaria. Orocariaceae. In many of the books, you will see that Orocariaceae have two genera. One is Orocaria, which is very common, again in our gardens, in colleges and parks, everywhere you can see them. Many call them as the Christmas tree, a typical cone shape or pyramid shape structure of the plant. And then you get agathis. The leaves are a little flattened in agathis. In Orocaria, there are two species. One is Orocaria cookie, where the leaves are needle-like, spiny, and Orocaria bidwilli, where the leaves are flattened. Many of the books you see, as I told you, there are two genera, Orocaria and agathis. But there is another new member found, Wollemia nobilis, commonly known as Wollemi pine. Wollemia, these are the leaves of agathis. You can see them broad. This is the leaf structure of Wollemia nobilis. Again, Wollemia nobilis was found as a fossil first, and then the living plant in the year 1994 by David Noble. From Wollemi National Park, Australia. New South Wales, Sydney. So another important characteristic feature and very important, like Metasequoia, which was found as a fossil first and then the living plant. We come to the next family, Cephalotaxaceae. Again, I was fortunate to see them in China. So these are the male cones and these are the female cones, the mature seeds. They have matured to form the seeds. Even I make the students work out in their MSc classes. So the leaves are quite flattened over here and they are very characteristic. They are very characteristic. Why I tell you that these leaves only can help in the identification of the genus, because these conifers, as you have already studied, as you have already read in the books, that pinus, they have the resin canals and all these things are there. There is transmission tissue. So the position of the resin canals and the transmission tissue is very characteristic, and thereby from the section of the leaves you can identify which species, which genus it is. Okay. The last family among the conifers is Taxaceae. Now, in the earlier classification, you will see that Taxaceae was placed within the coniferophytes. Taxaceae, along with Cephalotaxaceae, Taxaceae, not much of a difference, because why so named? Because similarity of the leaves. But later, we find that Taxus was separated from the conifers and a new order, Taxilus, has been created with the single family Taxaceae, and there are 5 genera. Common is Taxus. All have heard about it. So Taxus plant, you see the ovules which are there. So this was taken in Germany, in Bonn University, where we saw that there were all these trees there, and with these cones, the not cones, I would say, the ovules. Okay, and the characteristic feature is these red color things. This photograph you will see in the cover of Bhatnagar and Moitra book, Gymnosperm book, or BM Jodi and Chaya Bishar. I don't exactly remember, but they have this cover photo there with this Taxus plant. Now these ovules, they are arylate. This is the aryl which is so brightly colored. This is the aryl which is so brightly colored. And as you have already heard, that Taxus is very important. Now, because of this arylate ovule, which do not form the typical cones, that is why Taxus has been separated from the conifers and placed within this order Taxilus. This is the reason for separation of Taxus. Okay. So we find that almost all these conifers, as we have seen, that Taxus is important. Why? Because Taxus, as you know, Taxol, Taxus brevifolia, from where Taxol is obtained from the bark. And so they are very important medicinally. Otherwise, we find that these conifers, they are the timber yielding plants. You get your essential oils, resins, your drugs yielding plants are there within the conifers. So, and some food items also important as food. So conifers are there. Okay. We pass on to the next group. Sorry. Ginkgo biloba, the maidenhair tree, native of China. So in China and Japan, these are like roadside trees. This is the plant in the institute. There are a lot of them. So I took a photograph of this. There are many others. I felt this is good enough to give you an idea. So the leaves which are there, the bilobe leaves, Ginkgo biloba, as the name implies. These are the young ovules. You see that this stalk, the peduncle, it bifurcates. You see the diagrams in the book also, the two ovules which are there. And this is covered at the base by a collar. This is the characteristic feature. Two of them are there. And when they mature, these are the ovules which are there, which become seeds. So these start appearing by the end of March, April. By the end of March, this is in April when I took this, so they are a little larger. So by the end of March, they are much smaller, much smaller. And then by May, June, July, August, September, October, they become large. And these seeds are edible. The seeds are edible. And any of the food items, any delicacy, they always put this ginkgo seed. Or to honor any guest or anyone, special dishes are prepared, they use this ginkgo seed. But truly speaking, I did not like it. So these are the ovules. So again, a much closer view, you see the pollination drop. So well it has come out. This is the collar. And I have not put any sections of this purposely. So when we make a section, we see how the structure is there. You have the tent pole over there, and this is which is the structure of the ovule in ginkgo. So again, these are the ovules. The collar is very well represented over here. In many cases, we find that, which I observed, I used to go every day and see, many times I used to see there are 3 of them or 4 of them together. But then I gradually observed that no, they are there when young, but when they mature, there are 2 of them only, 2 of them. This is the male. They again form a catkin-like structure. And another important structure is these are the 2 sporangia, and the structure is over here, which is known where they are held together. This is the hump. So collar in ginkgo female and the hump in male is the characteristic feature of ginkgo, the reproductive structures. Now, ginkgo, just let me tell you that ginkgo is very important. There is an active principle obtained from ginkgo, gingolide B, which is medicinally very important. Gingolide B, which is medicinally very important, and it is used for the treatment of your, say, cerebral insufficiency or vertigo, all these diseases. Coming on to ephedra, we come into the Gnetophyta group. The Gnetophyta group is represented by 3 members: ephedra, Gnetum, welwitschia. So ephedra. And in this group, you will find plants which are trees like Gnetum, they are shrubs, or they are lianas. Most of the species of Gnetum, they are lianas. You have read in the books. I will come to it when we come to Gnetum. So ephedra, they are small shrubs, and you will get to see profusely these plants growing in Himachal Pradesh. So this is a photograph taken from there 2 years back when I visited there, and they were producing these male cones. There's a female plant. I did not put many so that I don't get not short of space, but they look similar. This is the male. From far you can see they are yellowish in nature, and the females, they are slightly orange in nature. And you see when they are enlarged, see these male cones. They are again composed of these opposite decussate manner arranged. These are the bracts. And the axil of the bracts, you find the male flowers. The female is almost similar, and you find the ovules are born. In males, almost all of them are fertile except the basal one or two pairs. And in the female, the lower ones are all sterile, the upper ones are only fertile. This is the dissected structure, which I usually show them in the class. Luckily, I had taken a photograph. So this is the male flower, each, and this is the sporangia. There's a stalk over here covered by the bracteoles. So this is the bract, these are the bracteoles. So the bracteoles, two anterior, posterior arranged bracteoles. So entire male cones or strobili and the female ones. So this is ephedra. From ephedra, we get ephedrine. Not all the species. Ephedra cynica, Ephedra equisitina, Ephedra gerardiana. And this is used for any cough syrup as a bronchodilator. So ephedrine is obtained from there. We come to netum. Most of the species of netum, they are lianas. A few are trees like Netum nemon. You get in the books, and it's a favorite question. Netum nemon is a tree, Netum ula is a liana. The leaves are angiosperm-like, so they have many angiospermic features. And you see the male cones. They are again composed of these number of bracts, which are fused together. These are the closed ones, fused together, forming cupulate bracts, collars, they are known as. And from the rim of it, you get the male flowers coming out, maybe in 3 or 4 rings. Contrast with the female, again you have the collar over here, or cupulate bracts, or connate bracts also known as, and you find a single row of ovule. This is the difference with the male and the female. And this is the ovules have matured into seeds. So netum. And the last one is welwitschia. Photograph taken from NBRI Lucknow. They have there in pots Welwitschia mirabilis, their only species. Only species. And these 3 genera, Ephedra, Netum, Welwitschia, in many of the books you will see that there is a single family, Netaceae, order Netalis, and within it the 3 genera. But later, these 3 genera have been separated as 3 families. So we have now families as well as orders. So we have Ephedra, Ephedraceae, Ephedrillus within the group Netophyta. We have Netum, Netaceae, Netalis, and Welwitschia, Welwitchiaceae, Welwitchialis. Only species. Welwitschia is characteristically growing in the dry, arid zones, native of Namib Desert of South Africa. So, Welwitschia mirabilis. Characteristic feature is that they have a long, deeply penetrating taproot. The stem is very, very reduced, and you can just see in the desert floor that these leaves coming out. Usually, 1 pair of leaf comes out during the whole life cycle of the plant, lifespan of the plant. Rarely 2. So these are the leaves that are coming out with parallel venation, cross connections are there, and the leaves have indefinite growth. So on the desert floor, you will find them rolling and rolling and rolling because of the presence of meristematic tissue at the base of the leaf. So because of that, they have this indefinite growth of the leaves. So this is all about, I don't know, I'm taking a lot of time. So this is all about this gymnosperm. Different types. I have tried to give you an idea, a glimpse of it, not going into the details, the structural features. Now, these gymnosperms, what I showed you is all extant plants. But as I told you that they have a long evolutionary history in the Carboniferous period, and there are many fossil plants. I have worked so many of the fossil gymnosperms. So there are fossils. Now, what are fossils? So let us peep into the world of fossils and see what these fossils unravel for us. I hope I will get 10 or 15 minutes, I suppose. Fossils. They are direct or indirect evidences of prehistoric life. Various ways they are defined, or they are remains of the past. Fossil word comes from the Latin word fordere, which means to dig. And paleobotany is nothing but the branch of science that deals with the study of fossil plants. So we get this is a Glossopteris leaf from our Gondwana sediments. This is again a fern leaf from Gondwana sediments collected, and this is a fossil of a Angiosperm leaf. Then where do you find fossils? Fossils are mainly found, as they are said, that they are dug up from the earth. So they are found in the sedimentary rocks, or in the limestones, or within the coal seams, which coal is nothing but a fossil fuel, a combustible rock, you can say, in diatomites, or within ambers. Now, when will a plant or animal turn into a fossil? The normal condition is that when an organism dies, it decays, the normal process. But when will it turn into a fossil? When we can remove the factors which help in the decay and decomposition. So there should be certain conditions present in the environment, in the sediment, which will help in the fossilization. And the most important is that decay should be prevented. And the fossils, the plant parts, should get embedded or buried in the sediments. They should be enclosed or in a protected water where there are lot of sediments accumulating. They should be rapidly buried, otherwise when they were exposed, they will get decay and destroyed. There should be low oxygen content so that the microbes can do not grow well. Then presence of high concentration of toxic substance must be present to prevent decay. Absence of strong water or air current so that the fossil, the plant parts are not carried far away, and when they are carried far away, they will be broken up into fragments to prevent all that. The depth of water is very important. If there is shallow water, it will be carried far away. There should be low pH, that means acidic condition, and the grain size of the sediments must be very fine. In addition to that, we must have the kind of tissue. If the plant, I will talk about plant fossils mainly. If the plant is made up of soft tissues, it will decay fast. So if there are hard tissues, if there are lignified tissues more in the plant body, it will be better preserved because the hard tissues are mostly preserved. And the intensity and duration of the destructive forces, that means pressure of the sediments. So how much pressure is there by the overlying sediments, that is also an important factor. Now, this entire process of formation of a fossil, right from the birth of an organism, then there is death and burial, and there is diagenesis and covered by the sediments and turning into a fossil, the entire process is known as taphonomy. So during this process, lot of changes take place, lot of transformation takes place in the fossil, and ultimately only certain parts remain in the fossil that we finally recover. Now, why study fossils? Why paleobotany? We can reconstruction of the plant groups, tracing the evolution of the plant groups, how the structures functioned in the fossils, plants, biostratigraphy and correlation. So, biostratigraphic correlation, determination of paleoecology, paleoclimate, paleogeography. Very briefly, I am saying all this. So, each is a topic by itself. So, fossils unravel all these things. Then how fossils are formed, just to give you an idea. So, there is death, they get buried, some of them will decay, they will get entrapped by the sediments, gradually the sediments will come and fall. So, deeper down they will get entrapped within the sediments, and then they become hardened, and we are there to recover them. What are the different types of preservation? Students have read in books that Schoff 1975 has outlined 4 modes of preservation. One is cellular permineralization, that means turning into a stone, simple way to turn into a stone. Qualified compression, orthogenic preservation, and diuretic or hard part preservation. Through cellular permineralization mode of preservation, we get a petrified fossil or a coal ball, very common. So, that petrified fossil may be siliceous, may be calcareous, or may be filled with dolomite, or it may be pyritic, limonatic, phosphatic, or it may be mummified also with an ice, cryopreservation, with an amber, resin embedded, I will show you some photographs, and with an amberite, which is urine embedded fossil. So, you get different types of these petrified structures. Through qualified compression, that means, as the name implies, it gets compressed. So, in cellular permineralization, you get a 3-dimensional feature, and this is very important because you can study the internal as well as the external features of the plant structure. Then you get qualified compression, where there is a compression fossil and an impression fossil. Orthogenic preservation, you get the formation of a cast and a mold, and diuretic preservation, hard part preservation, such as your diatomaceous earth, your bones, teeth, vertebral columns, mollusk shell, all these are hard part preservation. Modes of preservation give us some type of fossils. Besides that, we get other types of fossils also. Your chemical fossils, kerogen, biomacromolecules, DNA, lot of work going on in fossil DNA, biomacromolecules, and you get trace fossils or ichnofossils, such as burrows, tracks, trails. All these are trace fossils. Lot of work is going on this chemical fossil, biomarkers, biomacromolecules. Just to mention that we worked on a fossil. Fossil means a glossopteris leaf, and we could extract the biomolecules present over there. So a lot of these work is also going on. So I had collected some glossopteris leaves, and from there extracted these biomacromolecules. Then we come to body fossils, structures which are there in the form of microfossils or maybe megafossils. Microfossils which are very small, not visible with the naked eye, whereas megafossils which are large enough to be visible with the naked eye. Then what is the range? So fossils which are within the range of 200 micron, we consider as microfossils. Larger than that, megafossils, which may be in the form of compression, impression, petrifactions, cast and mold. Then there are subfossils, incomplete fossils, pseudofossils, like fossils, but they are not true fossils. They are not any organic compounds. They may be derived fossils, fossils which are found, older fossils found in younger fossils. I was working on some sediments, and so in the younger sediment, say tertiary, there were a lot of these Permian fossils. So these are known as derived fossils. Then coal balls, petrified fossils. Paper coal which is only formed from leaf cuticle, which is known as paper coal. Then there is amber. Amber is nothing but fossilized resin. There is index fossil. I will come to it now. Index fossil, which are marker fossils. And there is the phytoliths, which are nothing but silicified structures, which are opaline silica bodies found within the plants. A lot of studies are now going on with phytoliths. So phytolith study is more important in cases where you do not get any spore and pollen grains. Either from the plant body itself, phytoliths are studied, or from the sediments. Then the naming of the fossil plants is very important. As you know that where we can assign to a cannot be assigned to a family, we say it's a form genus. Which can be assigned, we say it's an organ genus. Say leaf is Glossopteris, root is Vertebradia, things like that. But later we find that this has been changed into a morphotaxon. Morphotaxon now is the concept of morphotaxon. So we do not call them as organ fossil or form genus or organ genus. And as you know, for naming of fossils, we have the suffixes. Whatever name you get, you can give, but these suffixes must be correct as per the rule of nomenclature. Say your leaf, it will be phylum or fern-like terrace, stem, dendron, xylem, it is xylon, microsporangiate structure, theca, ovulate structure, carpon, sperma, seed is testa, then spores, spora or sporites, cone is strobus, and pre-pollen or pollen, you can say pollinitis. Now, before studying fossils, one must be very clear about the geological time scale. So this is the international chronostatic time scale, and sorry, I did not put the recent one. Recently, they have given 2022. So there, it's almost the same, because there's not much, nothing changes there. So that I could not change it, rather did not change it. But this is the international chronostatic time scale, and all students must have an idea about the time scale. Now these are petrified fossils, again collected by me. So these are the fossil woods, and we cut them and then make a section and study them. I will show you. These are compression impression, again collected from our lower Gondwana coalfields. Many of the coalfields which I visit for my work. So you see the organic matter is present over here. So this is part and counterpart. When you break the sediment, you get these fossils like this. There is no organic matter, so you just get the impression of the leaf. So this is a negative, this is a positive. This is a impression fossil, Tylophyllum, which is an index fossil of your upper Gondwana, Tylophyllum. Tylophyllum is the leaf of Williamsonia seawardiana. So again, Glossopteris leaf, you see that some matter is present, again partly withered Glossopteris leaf. This is root of Glossopteris. This is Vertebradia, see Vertebradia, typical structure, and profuse you get in the coal mines. Again, Glossopteris leaf, the fern leaf, which we found, just to give you an idea how fossils look like. These are the other types of fossils, the stromatolite, the compression impression fossil, petrified fossil, the orthogenic preservation, you see the mollusk shells, ammonites they are known as, the dewey particle hard part preservation, and the pseudo fossil. So again, these fossils, amber, and you can see within amber, well preserved insects. There may be plant part also preserved. So amber, highly valued as a precious gem. Then there are trace fossils. So we had said what are trace fossils: tracks, trails, burrows, all these are footprints. Some marker fossils, Stylophyllum. Then you get Glossopteris, which is lower Gondwana. Middle Gondwana is Dicroidium. Upper Gondwana is Stylophyllum. Then they are reconstructed fossils. As you know that all fossils that we study, they are all reconstructed fossils. So it helps us in the reconstruction. So yeah, Williamsonia seawardiana, which was reconstructed by Professor Birbal Chani in the year 1932. And this is the gate of Birbal Chani Institute of Palaeobotany, which was known as earlier, now known as Paleosciences, where you get Williamsonia seawardiana. Another is your Pentoxylon. Pentoxylon, again reconstructed by Chani in the year 1948. Pentoxylon Chani. Again, there is a stone slab with all the petrified wood structures, just to give you an idea. Then how do we study fossils? We make a thin section, we make a peel, or we macerate. That is known as maceration. So when I was working in China, when on these petrified fossils, so these are the machines which helps in the cutting of the fossil. So these were the fossils that are there, huge fossils one. So cutting, then grinding, and then you make a thin section. All the fossil slides that you see in the class, they are all made by this process. So this was the structure. It's a plant which is Saroneus, a tree fern. Tree fern, it is known as Saroneus. So this Saroneus is characterized by a plant, tree fern found in the Paleozoic, that is during the Permian, where the stem is there and the entire structure, these are the root mantle. So all surrounding it is the root. So this is the section, not fully formed section. These are the fully formed sections. Okay, just to give you a glimpse of how they look like and how thin it should be made so that you can study the details. Another thin section, this is Avrainia. This is your Lygenopteris that you see in the slides in class. These are in your course. This is fossils Saroneus again, but this is a peel. We could take a peel from it. Sometimes it is not possible to take a peel, but sometimes peels can be possible, especially from coal balls. This is another fossil. This is Lepidodendron. Then how do we determine paleoclimate? Through study of tree rings, nearest living relative, leaf pattern, leaf physiognomy, or stomatal index. Stomatal index. So study of tree rings is dendrochronology, and we get a lot of information about the past climate. Then ancient DNA also. Ancient DNA first, 1990, when Goldenberg and colleagues, they isolated this DNA from magnolia plant, magnolia leaf. Now we come to the Gondwana. Everyone is very familiar with Gondwana, and we know that our supercontinent, which was Pangaea, surrounded by Panthalassa. Later, they splitted into the northern and southern hemisphere, which is known as the Laurasia and Gondwana land. But in the recent books, you will find that the supercontinent was known as Rodinia, which splitted into Western Himalaya, Western Gondwana, Eastern Gondwana, and Laurasia. And in between, you find the Tethys Sea. And this is the division of Gondwana. We have the bipartite or the tripartite division. Bipartite is lower and upper, tripartite is lower, middle, and upper. And then how the Gondwana sedimentation started with the deposition of the glacier sediments. And this is how coal deposition is there. So these are all coal beds, coal deposits. Coal you get either during the Permian period or during the tertiary period. So in our country, we have Permian coal, which is found in Raniganj, Asasol, Jhoria, Dhanbad, etc. And tertiary coal, you will get it in some pockets in Darjeeling, Sikkim, Arunachal, Bhutan, all these places. So these are the different coal fields. And then how coal is formed, plant parts get buried, and As they get buried, they are broken up into fragments. Fossils, as you know, that they are all broken up into fragments, and they form a sediment, which is coal is nothing but a organic sedimentary rock and is a fossil fuel. Then how do they, this was during my field work when I was collecting these material from bore cores. Before they set up a mine, these coding is done, boring is done, and from these bore cores we collect sample, and in the bore cores also you find such so good preservation of the fossils, fern leaf. Now I just want to touch that allied to paleobotany, there's another important branch which is palynology, study of pollen grains, and fossil pollen we say paleopalynology. And this I just want to show you that when I was studying with Saronia's stem, those slides that I showed you, within them, this was a thing, within them there were small structures, rounded structures. So careful observation, I found that they are nothing but coprolites, because coprolite study is very important. So they are coprolites, coprolites which are left within the stem by some insect. So, and these are the structures, and what they ate also, see the tracheidal elements, the cuticular structures, the spores, the fungal spores, wood fragments, they are all found within the coprolites. So it helps us to identify what the insect ate, what was the food habit of the insect, and the co-evolution during the time, how a plant insect co-evolve. And the scope of Palaeobotany Palynology to determine the past vegetation, climate, origin and evolution of the plants, paleogeography, stratigraphic correlation, and to determine when the fossil appeared first, and age also. I have said this very briefly because there is another important point is determination of the age of the rock. Through fossils, through dating also we can determine, and the applied aspect is exploration of fossil fuels, coal and petroleum, then palynostratigraphy, and defining ancient shoals. So just to give you an idea what paleobotany and fossils are all about. So thank you so much. And all fossils are found within the sediments of the earth, and we are only there to excavate them, recover them, and these fossils, as it is well written over here, that they are the rocks are the tombstones on which the buried dead plants, animals also, fossils are there, have written their own epitaphs, and we are there to unravel all the mysteries that they have left behind. So thank you so much for your patient hearing. I don't know how much I could convey, but anyway, giving you an idea about it. Okay, thank you so much.
Thank you so much, ma'am. You have conveyed a lot, actually.
No, this is in a very short time. I just tried to give you some idea about it.
Actually, there was a very excellent lecture.
Because this type of lecture, it is very much essential, and it is very important for the students concerned. Nowadays it is nowadays we do have to such type of lecture, and it is very rare actually this topic, gymnosperm paleobotany, in a very nice presentation, how the students can get the inspiration in this particular subject. Actually I call it as the red subject, and that's why, because it is the endangered right now, ma'am. So it presented very nicely, and our students and the participants will get much more inspiration from your very nice talk. Thank you, ma'am, on behalf of the Botany Department of Durgapur Government College. Thank you, ma'am.
Yeah, I tried to give you an idea, the students. I don't know how much I could convey to them.
Actually, teaching in this subject, now the teachers available in this subject for teaching in the particular basic botany of the gymnosperm paleobotany is now very rare, and that's why
very rare.
Actually, it is actually we long your long sustenance, ma'am, for the benefit of students, so that they can get a lot from you in future.
Uh,
Thank you, ma'am.
I have retired long time, but still they are not just leaving me. Every day I tell them I am just not
Yes,
possible to continue. But
yes,
again, for the benefit of the students. Thank you
yes,
so much. Thank you.
yes,
Thank you so much.
yes. We are so much grateful, ma'am. We are
Most,
so much
most,
grateful.
most
Mona,
welcome. Most welcome. Thank you. Most welcome.
Exactly, sir. I feel really nostalgic as it is reminding me my BSc and MSc days. So we got the chance to know about many unknown things. Like we had a lot of misconceptions. Like thuja we see here is not a thuja; it's actually biota. We didn't know
No,
that. We
no,
didn't
it is
know
not.
that
It is
actually.
not.
We have lots of misconceptions and. Studying about fossils and paleobotany is the next best thing because we do not know that whether we can use the time machine or not in future. But if we can study fossils and paleobotany in a proper way, then we can go to the past and see everything.
It is
Like,
interesting, very interesting. I tell
very
you,
interesting
not because
subject.
it's I deal with these subjects, but gymnosperms, pelibotany, any. Now, when we do fossils, we have to go through from algae to higher plants, algae to angiosperms, because you can get any fossils. So it's interesting. That much I can tell you.
Exactly, exactly. মানে like কি বলি আমি ভেরি মাচ মানে থ্যাঙ্কফুল টু ইউ
না,
আর
না, না।
এমনি কি ভালো
আমি
আছেন তো
চেষ্টা করেছি, খুব ভালো না, চলছে মোটামুটি।
আচ্ছা
চেষ্টা
আচ্ছা
করেছি কিছুটা বলার জন্য। ঠিক
না
আছে।
যে as
একটু সময় নিয়ে নিয়েছি বেশি, কিছু করার
no
ছিল
no
না
no no
আমার।
it was completely fine and it was a very interesting lecture. Thank you so much,
হ্যাঁ,
ma'am. Thank you.
বেশি সময় নিইনি, ১ ঘন্টা ১৫ মিনিট নিয়েছি।
Whatever you took, it was fantastic.
ওকে, থ্যাংক ইউ সো মাচ, থ্যাংক ইউ সো মাচ। ওকে, ওকে, and wish you all the best.
thank, thank you, ma'am. Thank you.
Okay.
Okay. Thank you, ma'am. Now the session is open for interactions, and we will take few questions if they have any. So I think there is no such questions as the topic ma'am covered was so much compact. So we will move to our next eminent speaker. Our next speaker is an eminent professor and scientist in the field of taxonomy. It is again my privilege to introduce that person whom we used to call the taxonomy storehouse when I was a MSc student. We often went to Sir's room and did a lot of trouble to him by asking so many questions regarding plant taxonomy and identification. But he never got annoyed and every time welcome us with a beautiful and calm smile. Please let me introduce our next speaker, Professor Shobhan Kumar Mukherjee, FLS London. Professor Mukherjee did his MSc from Department of Botany, University of Kalyani Nadia, West Bengal in 1975 and completed his PhD in 1992 from the same university. Professor Mukherjee has 40 years of teaching experience in PG course and 37 years of research experience. Professor Mukherjee has also research experience of more than 40 years. After retirement from University of Kalyani in March 2017, Professor Mukherjee was attached with the university as adjunct professor on the basis of special one-year appointment by the university authority. Now, Professor Mukherjee has been working as a guest faculty for teaching plant systematics in the Department of Botany, West Bengal State University. Professor Mukherjee started his career as lecturer in botany at Navadvip Vidyasagar College, then in APC College, New Barakpur, and finally joined the University of Kalyani 1985 as a lecturer in the Department of Botany and continued his service as professor in taxonomy of Angiosperm and biosystematics section till retirement 2017. Professor Mukherjee possesses his keen interest on the composite, especially for the sheep cellar morphology and sheep cellar anatomy. Other fields of interest in botany of Professor Mukherjee are confined with the study of floristic, ethnobotany, biodiversity, pharmacognosy, plant anatomy, and conservation. Professor Mukherjee is now guiding one research student. 22 students have already achieved their PhD degree under his able guidance. He has published 11 books and has edited 3 books jointly. More than 219 research papers of him are published in different reputed national and international journals. Professor Mukherjee was awarded Professor Y.D. Tyagi Gold Medal on 2017 by IAAT in the Department of Botany, University of Delhi, for his research contribution and collaborative research work with Swedish botanical science. He has awarded Linnaeus Jubilee Medal on 2008 of the Royal Swedish Academy of Sciences. Professor Mukherjee has been selected as a leading scientist of the world 2013 by the International Biographical Centre, Cambridge, England. Professor Mukherjee is acting as evaluator of many PhD theses and PhD viva voce of different Indian university and abroad. Professor Mukherjee is life member of 7 professional academic bodies since 1990 till date, such as International Society of Plant Morphologists, Indian Association for Angiosperm Taxonomy, founder member of Association for Plant Taxonomy, founder member of East Himalayan Society for Spermatophyte Taxonomy, Indian Botanical Society of Bengal, and Swami Vivekananda Botanical Club. So get ready for a beautiful journey towards taxonomy and enjoy the lecture by Professor Shobhan Kumar Mukherjee. Over to you, sir.
Okay, thank you, Anish, for your uh, introduction, and I am also thankful to the organizer, Ashok Bhattacharya and Prashanta Saha for inviting to deliver this lecture. I am also grateful to the principal, Dr. Devnath Palit. He is well known to me for many years. Now I shall deliver my lecture on the Angiosperm diversity of India for the students, mainly on the basis of students. Now I am presenting my work. Okay. Now it is visible?
Yes, sir, it is visible.
Huh?
Yes, sir, it is visible. Yes, sir.
Okay.
Yes.
My topic is diversity of angiosperm in India. I shall give a brief idea about angiospermic flora and angiosperms in India. First of all, I say few things. The India has great diversity floristically because due to different phytogeographical, different ecological condition and different climate, and these plants are needed for every body because they are the source of food, medicine, materials, and economic and cultural importance. And there are about 4 lakh specimens of vascular plants on the earth, and near about 10% of the more to be discovered. So there is a major area now remain for fulfilling of the research work in this field. Now I am showing, I know previously told the Indian flora is very variable due to mainly climatic change and topographic and soils, and India is the 10th largest country of the world and single largest of Asia after China. And it is total angiospermic plant and total number of plants in India near about 47,500 13 species of plants in India, and which represents about 11.4% of the known plants of the world, according to. And India, we know that India is one of the megadiversity countries of the world, and it has about 18,664 species of angiosperm according to World Development Indicator 2004, or According to estimation of BSI by Singh and Das in 2017, the number is slightly less, 18,386. And the total endemic taxa is in India about 4,303. So the number of angiosperm, such a number of angiosperm is greatly variable in Indian context, and the total number of angiosperm of the world near about 350,000. Now the present estimate shows these angiospermic plants, 8,864 plants. These are distributed in 315 or more families according to concept of the family. According to Takhtajan, the number is greatly increases, but according to other authors, Conquest and others, APG, the number is slightly less, and the number of genera is near about 4,000. And what about the endemism? Percent of endemism, the phenomenon by which the taxa is restricted in India and different region, and this phenomenon is termed as endemism, and the taxa can be termed as endemic. And the angiospermic taxa, the endemic concentration varies according to different author. Near about average, and the percentage of endemic concentration is 28% in India. And I shall show some bird's eye view of the angiospermic flora of India. Because I am delivering the angiospermic flora, that's why these are the total landline, coastline, and these are the boundaries. This is not important for angiosperm. Just we know megadiversity, one of the 17 megadiversity country of the world, and India is one of the megadiversity country, and its position is previously it was belong to 11th position, but at present it is 10th position, 10th megadiversity countries, and each megadiversity country has more than 10,000 endemic species total. And the features of megadiversity countries is endemism at the level of species and general level. And megadiversity country, according to definition, the must contain 500 species of endemic plants and must borderline ecosystem, and both these characteristic features fulfilled by the Indian continent. That's why India belongs to megadiversity country. And these are the megadiversity countries and number of flora, number of angiosperms. First, Brazil, and in its position previously 11th position, but at present it is 10th position on the basis of number of taxa, 18,664. And these are the other countries, second Colombia, third China, Indonesia, Mexico, etc. This is the first Brazil, and India is 10th position on the basis of megadiversity countries. And these are the, initially there are 20, first time there are 20 megadiversity countries. These are the first installation, there are the 20 megadiversity countries, and after that megadiversity, these are the megadiversity countries of India. Now I am showing the biodiversity hotspot area. The hotspot area, there are 25, initially there was 20.
Excuse me sir, আমি স্যার অশোক বলছি স্যার। স্যার স্যার বলছি কাইন্ডলি একটু ওই আপনার স্ক্রিনটা একটু ফুল স্ক্রিন করে দিন আমাদের, এই তাহলে পার্টিসিপেন্টদের একটু দেখতে সুবিধা হবে। ওই স্ক্রিনটা স্যার একটু ফুল স্ক্রিন করে দিন কাইন্ডলি আপনার ওই প্রেজেন্টেশনে। হ্যাঁ ফুল স্ক্রিন করে দিন স্যার কাইন্ডলি।
দাও, হ্যাঁ ঠিক আছে। এটা কি হচ্ছে না? তাও তো হচ্ছে না, এটা কি করে করব বলো?
নিচের দিকে,
হ্যাঁ নিচের দিকে,
নিচের দিকে ডান দিকে দেখুন, নিচে টাস্ক বারে ওইখানে একটা
ও হ্যাঁ এটা
ওর পাশে না, ওর বাঁদিকে একটু ওইখানে ওই 100% যেটা আছে ওইখানে না, তার পাশেই যে সাইনটা দেওয়া আছে ওইটাতে, ওর ঠিক পাশেই, ওর পাশে, হ্যাঁ ওইটা করলে আপনার, হ্যাঁ স্যার, হ্যাঁ স্লাইড মোডে চলে যাবে। আপনি ওই আপার কি লোয়ার কি দিয়ে পরেরটায় চলে যেতে পারবেন।
থ্যাংক ইউ স্যার, থ্যাংক ইউ স্যার।
ঠিক আছে, আমি ঠিক বুঝতে পারলাম। হ্যাঁ, ঠিক আছে। না না না, ঠিক আছে। হ্যাঁ, না না মেঘা, ঠিক আছে। হ্যাঁ,
থ্যাংক ইউ স্যার।
হ্যাঁ, এই biodiversity hotspot, we know the India has 4 biodiversity hotspot region, and initially the condition for biodiversity hotspot, that the number of endemic species is 1,500 and 70% habitat loss already, and these are the 2 criteria for the hotspot regions. On the basis of this criteria, there are the 25 hotspot initially developed: the Western Ghat, Indo-Burma hotspot, and Sundaland. But after that, 9 hotspot again recognized by the World Authority in 2005, and total the number of hotspot 34. Previous installation, there are 3 hotspot Indian region, and the Himalayan region in the next 9 hotspot belongs to 9 hotspot region, and the biodiversity hotspot regions, 34 hotspot region. According to Myers, the first identified 10 tropical hotspot. And what are the condition for hotspot? There are 2 criteria. There are the many criteria, but out of these 2 are important for hotspot. Must contain at least 1,500 species of vascular plants as endemics, and it has lost at least 70% of its original vegetation. These are the 2 criteria for hotspot region, and the India has 4 hotspot region. These are the Himalaya, Western Ghats, Sri Lanka, Sundaland, and Indo-Burma. And out of these, these are the regions following. These are the regions belonging to Himalayan region, Pakistan, Tibet, Nepal, etc. Indo-Burma, Assam and Andaman group of islands, Northeast India excluding Assam and Andaman group of islands, Myanmar, etc. Western Ghats includes the entire Western Ghats plus Sri Lanka. Sundaland includes the Nicobar group of islands, plus Indonesia, Malaysia, etc. And now I am showing some hottest hotspots. NIAR again devised a new definition regarding hotspot region, considered the hottest hotspot, and in the endemic concentration is 0.2% of the global total and 70% of the habitat already lost. And according to him, these are the hotspot region: Deang, Debang Valley, Nokrek region. These are the hottest hotspot region, but these are not internationally accepted by the major scientific world. And the biodiversity hotspots, the Himalayan region. These are the Himalayan region, and now because I am showing. Hello? Diversity and endemism. The endemism is highest in Indo-Burma region. These are the plants, and endemism 51.9%. And the next and first is Sundaland 60%, and third is Western Ghats and Sri Lanka, and fourth is Himalayan region, 31.6% endemism. These are the endemism. Biodiversity hotspots of the world. Previously, I saw there are 34 hotspot region of the world. 34. These are the 34 hotspot region. But in 2018, there is another new hotspot recognized by the World Authority. These are the Yangtze Basin. Yangtze Basin. This is the Yangtze Basin near China and part of China and other parts adjacent to China. These are the new hotspot region according to World Authority. And there is another hotspot, and this another place recognized qualified for hotspot region. These are 36 is the North American coastal plain. These are the North American coastal plain. This is the another hotspots according to number of taxa and the habitat loss. This region again is qualified for hotspot region at 2020. And we know the phytogeographic regions of India, according to these are the common divisions according to phytogeographical regions of India. We know very well the Clark, Hooker, Chatterjee. These are the previous concept, but according to most latest by N.P. Balakrishnan in 1996, they have classified the Indian phytogeographical regions into 11 phytogeographical regions: Eastern Himalaya, Northwest Himalaya, Indo-Gangetic Plain, Assam, excluding Arunachal and North Bengal. Central India, arid zone, northwestern Ghat and northwest coast, southwestern Ghat and southwest coast, including Lakshadweep, and Deccan, eastern Ghat and Coromandel coast, and Andaman and Nicobar. Total, there are 11 phytogeographical regions at present. After that, anyone scientist has not classified India into. These are the phytogeographical zones of India. These are the number of taxa, number of area, and the percentage of endemism in different taxa. These are the regions, subcontinent. These are the phytogeographical. Already I have told the 11 phytogeographical regions. These are the 11 phytogeographical regions I previously mentioned, and the species diversity. Next, I am showing some species diversity of India. As because I am only talking about Angiosperm, the total number of world genera is, I previously told there are about 350,000 average, 350,000, 352,000 total species and 14,000 genera, and out of these India has about 18,664 genera and species and 2,984. These are the angiospermic, and these are the other groups of plants. And total, the number of flora in India is near about 46,000, 5,000, 46, 47, near about 47,000, about, and about 11 to 11.4% of total area. And I am showing some few diversity of as because I am discussing about all the phytogeographical regions. It will take half an hour. That's why I am showing some major differences between Eastern and Western Ghats, Eastern and Western Himalaya and Western Ghat. The floristically rich in Eastern Himalaya, these are the area, and the Eastern Himalaya there are 4,000 species of flowering plant, among angiospermic species endemic 1,800 is endemic. And the diversity, there is a great diversity of orchid, bamboo, rhododendron, quercus and hedychium. These 5 genera has great diversity in Eastern Himalaya, which are not recorded in Western Himalaya as well as Western Ghat. Whereas in Western Ghat mainly includes this portion, the number of taxa near about 4,000, and the maximum diversity of, we know the maximum spices of India are produced from Kerala and other parts, other adjacent state, and these are the Kerala is the major spice yielding plant. That's why the maximum diversity of spice, legumes and bamboos. India represents about 221% of the flowering plants of the world. According to Hooker, we know the J.D. Hooker, he worked on Indian flora mainly, and the flora of British India by J.D. Hooker. And Hooker in his literature mentioned the Indian flora is more varied than that of any other country equal area in the Eastern Hemisphere, if not the globe. That means India is one of the richest country having the great number of diversity, according to Hooker. And these are the dominant angiospermic family. The dominant angiospermic family, the number of India, the number of dominant angiospermic families is near about The Poaceae, Fabaceae, Orchidaceae, Asteraceae, and Rubiaceae. These are the number of dominant five dominant families in India. According in belonging to monocotyledon, Poaceae is the dominant, whereas Fabaceae among the dicotyledons, Fabaceae is the dominant, and other dominant family Orchidaceae, Asteraceae, and Rubiaceae. These are the number of taxa in respect to in India as well as in the world. These are the six: Cyperaceae, Euphorbiaceae, Acanthaceae, Rosaceae, Lamiaceae, and Scopulariaceae. These are the number of taxa belong to each family. These are the number of biogeographical region. These are the taxa, total area, and the number of species in different biogeographical regions. I am not discussing about the detail about the biogeographical regions. These are the taxa found in different biogeographical regions. We know the Esting Angiosperm. Esting, these are previously these plants has been recorded from Indian subcontinent by the previous author, but these are not available at present. That's why this is the Esting Angiosperm species from the wild in India and other regions. These are the Calantha whitiana, Orchidaceae, not reported from Sikkim, but Carex ripanda. These are the from Cyperaceae from Assam and Meghalaya. Previously, it has been recorded from Assam and Meghalaya region, but there is no these taxa at present. That's why these are the extinct angiosperm. These are the some other angiosperm, Corypha, Teleida, Asteraceae. These are the now of the many of the family, many of the genera which has been extinct from the Indian region. These are the other genera, and I am saying about another genera, Pleone leginaria, belonging to Orchidaceae, extinct from Meghalaya region. There is a journal published by the East Himalayan Society, which is known as Pleone, from the genus Pleone, a genus from Orchidaceae. The name of the journal is Pleone, published from East Himalayan Society, North Bengal. And this is actually the name of the genus from which the name of the journal is taken. We know that there are the two monocotyledons and dicotyledons. Monocotyledon and dicotyledons. The percentage of ratio of monocotyledons is 1:2.3, 1:2.3, similar to genus to species 1:4.7. These are the ratio of dicotyledon taxa and monocotyledon taxa in India. And some of the dominant genus, Impatiens, maximum number of species, 205, Primula, Carex, Dendrobium, Habenaria, Peticularis. These are the some. So, maximum number of species has been recorded from Impatiens. বেঙ্গলি নেম দোপাটি ওই ইয়েতে সব থেকে বেশি স্পিসিস পাওয়া যায়। There are over 60 families are monotypic. Monotypic, that means হচ্ছে number of genus one, maybe number of species may be more than one, but the number of genus must be one. This is termed as monotypic. There are about over 60 families in monotypic, and 33 families are typically monotypic. That means it has single genus and single species only. As for example, Tetracentaceae, there is only the vesselless angiosperm in India, Tetracentron sinens, found in Western Himalaya. Similarly, Succester, there is a single species, Succester agrestis. It is found in Eastern Himalaya. And the number of highly valuable timber belonging to 15% tree species are highly valuable for the timber production. These are some of the families which produce maximum number of timber, Meliaceae, Verbenaceae, Dictycarpaceae, Fabaceae, Lauraceae, etc. And these are the cold desert taxa. That means they form a cushion forming plant. The cold desert taxa, they form. I shall show these all some of the plants after some time. These are the cold desert taxa. Thylacospharum, Sasuria. These are the cold desert taxa. These are found in cold desert region of Kashmir, Himalaya, and other regions. And some of the curious plants in India, Nepenthes khasiana, no, Sapphire himalayana. This is the largest flower in India. This is the largest flower in India, belonging to Rafflechiaceae. We know that Rafflesia arnoldi is the largest flower of the world, and belonging to this family, there is no Rafflesia arnoldi, but there is another genus, Sapphire himalayana. This is the largest flower in India. These are the Belanophora. I shall show all the taxa after that. These are the curious taxa. Insectivorous plants belong to the maximum number of insectivorous plants belong to Lentivillaceae, Drosseraceae, and Nepenthes. Only one. And Nepenthes shows the disjunct type of distribution. That means it has more than 70 species, and 70 species. One species found in India, Nepenthes khasiana, another species found in Sri Lanka, and Madagascar also found in 20 species, and rest of the species found in southeast part of the world. That means it has the great diversity of Nepenthes as a disjunct family. Parasitic family, we know the Orobanchaceae, 54 species, Loranthecae and Cuscutaceae, Cuscutaceae, Santalaceae, Melanophораceae, Rafflechiaceae. There is only species Rafflechiaceae, Sapphire himalayana, parasitic family. Now I am showing some root parasitic plant, Agenetia indica. It is all belonging to Orobanchaceae, the root parasitic plant. There is the Balanophora daiica. It is the root. These are all the root parasitic found in Assam region mainly. The Assam region, and one of the plant Mitrestemanum jetamonsi, a root parasitic plant. It has been described as a new species, newly reported, but in 2018 it has been also recorded from Philippines. That this is not endemic. It has been also recorded in 2018 from the Philippines. That's why it is not endemic. It is a parasitic, root parasitic plant, but it grows on the Fagaceous plants, association with Fagaceous plants. Another family of Balanophораceae, it has been reported from Namdapa Biosphere Reserve in Arunachal Pradesh. Newly reported plant from India. Now I am showing some stem parasitic plant, Cuscuta chinensis. We know it is found in everywhere in Bengal. Dendrophthis falcata also found in Bengal, and Viscum orientale. These are the common stem parasitic plant found in Bengal. And now I am showing one of the smallest parasitic plant of the world. These are the Arceuthobium minuscetimum belonging to Santalaceae. These plants found in Himalayan conifers, mainly Pinaceae and Cupressaceae, and it is the smallest parasitic plant of the world. Arceuthobium minuscetimum found in Western Himalaya mainly. These are the plants, parasitic plants. Now I am showing the largest flower in India. Previously I told Rafflesia arnoldi, the largest flower, which has not reported from India. It is from Malay Peninsula. Similarly, there is another plant of the same family, the Sapphire himalayana, a root parasitic plant, reported from India, mainly from Arunachal, Nagaland, and this is the holoparasitic plant. The size of the flower is 15 to 30 centimeter across, one of the largest flower. saprophytic plant, the Haplothismia, these are the Balmaniaceae, Epipogium, Orchidaceae. These are the some saprophytic plant found in Indian continent. The aquatic plant. Now I am stating about some aquatic plants of India. There are the two type of classification. I shall omit the first one because it is to some burden for the student. That's why I am showing the simple version of the aquatic plants. Aquatic plants, aquatic angiosperm, they are the 2% of flowering plant, known plants habitat in, and 50% are represented in inland subcontinent. And these are the families of aquatic plant families in India. Podostomaceae, Lamiaceae. We know the Lamiaceae, Alismataceae, Nymphaceae, Aponogetenaceae, Nilamonaceae, etc. Ceratophyllaceae, Pontediaceae, Typhaceae, etc. But most of the students don't know about Podostomaceae, and this is a unique family in Angiosperm because this plant shows sometimes algae-like character, sometimes bryophyte-like character, sometimes pteridophyte-like character, and it is unique, and there is a and this is unique because this family only grows on free-flowing rocky substratum. That's mean this is not available in the stagnant water. It is found on the South India free-flowing rocky substratum. This grows by a special organ termed as Haptera, which is found in the members of Podostomaceae. This is a unique family Angiosperm, which are not, but other families are quite different from Podostomaceae. These are the some figures of Podostomaceae, sometimes bryophyte-like, sometimes selaginella-like, sometimes algae-like. These are the different morphological forms of Podostomaceae. And one thing, if the Podostomaceae grows in aquatic condition in any area, that means it is clear of pollution because it is the indicator of pollution. In polluted water, these plants will not grow. According to growth form, the aquatic plants is different types. I shall omit this one because it is to some difficult for the general student. That's why I am passing to another form of classification. Aquatic vascular plant on the basis of habitats. We know the free-floating hydrophytes. Free-floating hydrophytes. We know the Iconia cassipes. Iconia cassipes. It is free-floating hydrophyte. Surface water in contact with air and stagnant water. Free-floating hydrophytes. Suspended hydrophytes anchor and submerged in the young stage and lost their root, and so they cannot floating water restricted in stagnant pond and take. Usually the Ceratophyllum demersum, the most common, Jhaji. It is available in any pond in India and area colon. These are the Hytila. These are the other plants of this category. I am showing submerged anchored hydrophytes. These are grow in anchored condition, but and these are found in stagnant and running water also. Most common in West Bengal is Aponogeton natans, Vallisneria cristata, etc. These are the Aponogeton natans. Anchored hydrophytes with floating leaves. Anchored hydrophytes with floating leaves, we know belong to the Nilambonucifera, Polostemon sagittaria. These are the Sagittaria. And emergent aquatic hydrophytes, plants anchored to substratum underwater, but produce aerial shoot projecting well above water. And these are the different plants belong to this family: Bacopa moneri, Brambri, Limnocharis, Monocaria, and Acorus calamus. Acorus calamus means such botch. It is very important medicinal plant. Roots are used as medicine. Anchored hydrophytes with floating shoots. We know that Ipomea aquatica, Trapedominensis, and these are the different groups of plants. I am showing some groups of plants, and again showing some interesting plants of India. Nymphia rubra, this is a native of Bengal. It has a native of Bengal. এবং this plant produce up to 5 flowers at a time. Limnophila indica belong to Plantageneaceae. Previously belonged to another family. This is the important medicinal plant, antiseptic and cured dysentery. These are Spirodella. Commonly we know the different ponds covered with Spirodella polylysa. And one thing, these are not usually occur. This is the Nuphar lutea, belong to Nymphaceae. Previously Nymphaceae, and this is the trimerous flower, like other flowers of primitive dicot having trimerous flower. Actually the dicot is tetramerous or pentamerous, but primitive dicots are trimerous. It is trimerous flower. Lemna major, we know very well, found in everywhere in water. And Wolffia microscopica, this is the smallest free living plant. Previously I told the smallest parasitic plant Arceuthobium mycoscipium belong to Lorenthaceae, and this is the smallest free living plant, Wolffia microscopica. It is also found in India and Bangladesh also. Wolffia microscopica, these are the different parts of. And we know that another important plant, aquatic plant, is Urial ferox, belonging to Nymphaceae. Previously belonged to Nymphaceae, but at present Urialaceae. The seed are very edible, and these are exported from India. Roots, rhizomes are eaten by the local humans, and seeds are exported from India. And these are the primitive genera in Indian family. There are many primitive families in India. According to Takhtajan, considered the Northeast India and Assam region as the cradle of flooding plants. Cradle of flooding plants, that means মানে দোলনা, বাচ্চাদের যেমন দোলনা থাকে তেমনি ক্রেডল অফ অ্যাঞ্জিওস্পার্ম, মানে অ্যাঞ্জিওস্পার্মের দোলনা। তার মানে হচ্ছে এখানে সব প্রিমিটিভ ফ্যামিলিগুলো পাওয়া যায়। There are in this region number of primitive families হচ্ছে 131 primitive angiospermic species are found, and some of the primitive family Magnoliaceae, Tetracanthaceae, Anonaceae, Mysticaceae, Cisandaceae, these are the family, and some of the genera. These are found in the Northeast India and Assam region. That's why Takhtajan considered this region as the cradle of angiosperm flowering. Angiosperm, that means angiosperm evolved from this region. This is the primitive region, and now I am showing some few about mangroves. We know these are the mangroves. They are the 7% of the world mangroves found in India, and the maximum mangroves are found in Sundarban area, and other also found in other maximum mangroves are found in Sundarban and Assam and Nicobar Islands. Other mangrove containing states are Andhra Pradesh, Tamil Nadu, Orissa, Maharashtra, Gujarat, Karnataka, etc. And here some of the dominant mangroves, Bugera gymnorrhiza. We know I shall show some figures of kankra, Bengali name kankra, Bugera Heritiera forms, Sabi, Acanthus ilicifolias, Herbaceous halophytes. Some these are found in sea beach of mangrove region, Susubium, Pseuda, and Salicornia. These are the three genera dominantly found in the sandy beach of mangrove region. And we know there are only 2 palms have been recorded from Indian mangrove region. One is Naiva fruticans and Phoenix paludosa. Only these 2 mangroves are found in Indian region. Now I am showing some figures. Heitera literalis, Nipa peticans, Phoenix paludosa, Henthal. This is Henthal. Golpata. This is Nipa peticans, termed as Bengali name Golpata. Phoenix paludosa, it is also known as Henthal. Chansadagar use this stem for destruction of the snake. Sundari, Heritiera forms. Acanthasis leucifolius measures survey plants in mangrove region. Kakra, Bugara gymnorrhiza, showing viviparous germination. Rhizopora, this is the root characteristic features of the mangrove plants, Rhizopora. Now some dicotyledonous family. The dominant dicotyledonous, these are the dominant dicotyledonous families. Asteraceae, Fabaceae, etc. These are the dominant dicotyledonous family. And out of 203 dicot families, 10 families, 40% genera, contain 40% genera, and 42% species. And the rest of the 193 families, only 84 families and monogeneric. Most of the taxa are monogeneric. And now showing some monocotyledonous taxa. These are the dominant monocotyledonous taxa: Poaceae, Orchidaceae, Liliaceae, Cyperaceae. And out of 43 monocot families, 7 families includes 87% of the total monocot genera and species, and remaining 39 families include only 13% of the monocot genera and species. That means maximum number of taxa belong to only 7 families. Largest, previously I showed the largest genera in India, and out of 189 monotypic genera and 100 out of monotypic genera found in India, out of the 133 dicot and 56 monocot. Diversity of endemics. There's another part of the Indian flora, the diversity, the endemics. Endemics, that means these are restricted in India region. And there is a very controversy regarding the endemic taxa in India. And average number of endemism concentration in India is near about 28%, according to Singh and al. According to that, the number of endemic species is 5,600, and the number of endemic mainly restricted in Himalayan region and peninsular India, and few are also found in Andaman and Nicobar region. These are the other, according to recent estimation. According to recent estimation, the number of endemic taxa 4,045 and 155 families, and these are the maximum in dicotyledons, and these are the number of genera in dicotyledons, and these are the monocotyledons taxa. Now I'm showing some endemic concentration. Previously I told the average different author estimated, but average at present the 28% Indian flora is endemic, and the number of endemic concentration according to different author is greatly variable. Now 33%, Rao 33.5, 33% by Rao, 36 by Balakrishnan, 40% by Maheshwari, and 50% by Chatterjee, and they have estimated only the dicots flora, not in all Angiosperm. They are the some of the top dominant Magnoliopsida, that means it's a dicotyledons and monocotyledons family. According to modern concept, the dicot is known as Magnoliopsida and monocotyledopsida. These are the number of endemic taxa in different family. Leguminosae, these are the some taxa and number of belonging to Magnoliophyta and belonging to Liliophyta. Endemic angiosperm, distribution of endemic angiosperm, maximum endemic angiosperm found in this Western Ghat, endemic angiosperm Western Ghat, Northeast India and Eastern Himalaya. They are the Western Himalaya. These are the 4 zones which having the maximum number of endemic taxa, and total number of endemic taxa is 4,353. And the endemic number of endemic genera. Endemic genera, previously it has been estimated as 142 endemic genera in India according to Sarkar, and 148 endemic genera by Nayar. I have estimated and reduced from 142 to 121 endemic genera by myself and my student. Dr. Mitro, and at present there is latest estimation by Arduin, Irwin, and Narasimham from in Madras Christian College. They have published a paper which has 49 endemic genera. Which is the reason for reducing the number of genera? There are three reasons for reducing. One is the genera previously recognized as endemic genera, but it has been merged with another genera. That's why the number is decreased, and the genera previously designated as endemic, but it has been recorded from outside India. So the status has lost, and so it is reduce the number of genera and increases the number of genera as because there is a new taxa described by Indian author, and this is the newly endemic recognized. These are the some of the peninsular India according to Narasimham, the number of endemic genera, 49 endemic genera, 40 peninsular India, 4 round in Himalayan, Indian Himalaya, 3 in Andaman Nicobar Islands, 1 peninsular and North India, and 1 is disjunct distribution. 1 is disjunct type. These are the category of endemic taxa. Peninsular India and Nicobar Islands. These are the some peninsular India. Some 80 type belong to 36 unified specified. These are the some endemic genera of peninsular India. There are the maximum number of peninsular India, and these are the peninsular 40 endemic genera: Adenone asteraceae, Hubardia poaceae, and one of the endemic genera is Karnataka. It was created by Mukherjee and Constance. Constance is World Authority of Apiaceae, and Professor P.K. Mukherjee, that means it's Calcutta University professor, ex-professor, Professor P.K. Mukherjee. He had created these two genera, Karnataka and Bonosisova, by Mukherjee, and Constance is the World Authority of Apiaceae, and the Willissia porostemaceae. The Himalayan region, there are the 4 endemic genera in India. The name of the genus is India: Orchidaceae, Kashmiria, Scopulariaceae, and Paracampia, and Stabularia. Andaman and Nicobar Island, this Nicobaridendron, Pseudospora, and Sphaanthera. These are the 3 endemic genera found in Andaman region. And peninsular India and North India, only endemic genera is Hardwickia, belonging to Fabaceae, and among the genera, only Bentinkia shows the disjunct type of distribution. Disjunct, that means it has been reported from more 2 widely separated geographical region. Bentinkia condappana, it is found in Kerala and Tamil Nadu, and Bentinkia Nicobar Island. That means these 2 regions are widely separated, and these 2 species found in India. This is a disjunct type of distribution in India. Some of the monocotyledons and dicotyledons, these are the highest number of endemic taxa in different groups. Now I am passing, these are not these are the number of endemic genera according to myself and my student estimated the number of endemic genera highest in Poaceae, whereas other 2 authors estimated Acanthaceae has the highest number of genera. Similarly, these are the endemic genera in Himalayan region and Assam region. According to my estimation, the number of these are the found in Himalayan region. These are the Kedarnatha, it has been also created by Mukherjee, Constance Edgaria. These are the endemic genera. I am showing some photographs, not all. Endemic in peninsular India and Western Ghat. According to my according to estimation, according to estimation of myself and my student, these are the endemic genera of India. There are the many endemic genera. The names are not recognizable easily. That's why I am passing through the number of genera. These are found in, according to my estimation, the number of endemic genera only 10 in Andaman and Nicobar Islands. Previously, I saw why their number is gradually decreases. Their number status due to new distributional area outside India. It is the one of the reason. Colibrochia. Previously, it was restricted in India, but it has been also reported from Pakistan and West China. So it is now not endemic. Hemigraphia. It is copulaceae. It has been reported from China. That's why the number is gradually decreases. And another reason for number of decreasing is SK Mukherjee. SK Mukherjee created by Malik and Sengupta, previously named as endemic genus, but this genus has been merged with Phagopyrum. So the status, this is not at present recognized as endemic. Previously, we know the earlier classification, the Nilgiria anthus is the highest genus having highest number of endemic species. There are about 20 endemic species in Nilgiria anthus. 20 endemic species in Nilgiri anthus found in South India. But at present, the genus has been transferred to the genus Strobilanthus. So this genus is not endemic. These are the some Santapao created by Balakrishnan and Subramaniam. It has been also transferred to Hygrophila. So it is now endemic. This is the reason for decreasing the number of endemic genera. And new endemic genera, before my publication, these are not included in India. These are the some of the genera which first time I have introduced as endemic in Indian list. Recent endemic species, these are the highest number of endemic species, Barbadeaceae, Himalayan families, the peninsular families, these are the endemic percentage. Endemic in different states of territory, the number of endemic states are Arunachal maximum number of endemic taxa, Kerala, Jammu and Kashmir. Yes, I shall gist in. These are the. I am showing some endemic taxa. Sanotis concolens is endemic. These are the endemic taxa in India. These are the endemic taxa. All are endemic in Indian region. Pinda concolens. This is the wild flower, but it can be used as ornamental flower at present. And the Shivaji University has created these plants as a ornamental plant from the wild plant, and they have propagated in their garden, and the seeds are distributed to the farmer for ornamental purpose. The Delphinia malabaricum. These are all are endemic. Monosusova. Now, Willicia selaginella, that means I told this the Podostomaceae like selaginella like structure. That's why selaginella is. These are the endemic taxa. Kashmiria. Edgaria darjeelensis found in Darjeeling area. SK Mukherjee, now it is not endemic. It is belonging to Phagopyrum esculentum. Previously endemic. Chandrasekhar belonging to Podostomaceae. Belonging to Poaceae. Previously, I told the Bentinkia, there is two endemic disjunct type of distribution. One is Bentinkia condappana. It is found in Agasthamala hills of Kerala. And these are the plants found in Agasthamala hills. This is the Bentinkia condappana found in Agasthamala hills. This is the structure of Bentinkia condappana. and this is Bentinkia nicoborica found in Nicobar Island. This is the only endemic genus Tobalanthus. Some curious plants, Tetracentrum cyanide vesselless angiosperm found in eastern Himalaya, which and this is one of the very interesting plants in angiosperm. This is Kingdonia uniflora. Kingdonia previously belonged to Sarcestes. These plants show like fern-like free dichotomous venation, which is not found in any other angiosperm. It is similarly Sarcestes agrestis. It has also shows the free dichotomous venation like other not in other dicotyledons. This is the largest inflorescence in India, from 26 below. We know this plant is Stobalanthus kunthiana kunji, নীল কুঞ্জিরি এটা হচ্ছে it grows after 12-year intervals in South Indian hills. And when these plants produce flower, all the foreigners come to see the flowers. And we know that rhododendron means this is a large, but this rhododendron, the only species in India 2 to 3 centimeter high, rhododendron nivuli. And according to Hutchinson, this is found in Himalaya, northeast India, and this Magnolia pterocarpa, according to Hutchinson, this is the most ancient species of living angiosperm found in. And the Chistole Himalayensis, this is the highest recorded altitudinal plants of the world. It has been recorded from 21,000 feet from Mount Comet by DJ Joel at Himalaya. It is the highest altitudinal recorded plants of the world, which has been recorded from India. And some of the other recorded high altitudinal plants. So you know the Sasoria Oholata Bombo Kamal. It is also found in Himalayan region. It is a state flower of Uttarakhand. The cushion-forming plant Thylacospasmum terephalicea. Some of the plants. Griffithella is another porostimacea found in Barfat Lake. It is found in South India, endemic in South India. It is the tallest orchid in India. And the Pinguicula alpine, now only insectivorous alpine found in alpine region. This is the species. Demes winteri, the vesselless angiosperm. We know the tulip tree. Our tulip plant is a small herbaceous. There is in Ranunculus there is a tree which is known as tulip tree, Liriodendron tulipifera. It is found in India. And Rheomimedi, Actia species in Ranunculus, the number of carpels is more than one, but it has only one carpel in Ranunculus. We know the Pereschia grandiflora. We know the cactus, there is no leaf-like structure, but in this structure, the Rhodocactus grandifolius, present name, it has distinct dicot-like leaves. And showing the Nilambo nucifera and the Nilambo poddo. The Botanical Survey of India, after 50 years of completion, they have published revised the name of his journal, Botanical Survey of India, Bulletin of the Botanical Survey of India, now as Nilambo from 2008. Few invasive flora. Know that these are the plants, Vachelia farnesiana, previous name Acacia farnesiana. Some of the exotic plants, it is very common. It is Cinedella, like Cinedella nodiflora, but the stalked flower found in everywhere in Bengal. Acanthosperma hispidum. These are the some. In most of the books, these are written as cotton bone plant dianam, but it is actually it should be bone plant dianas in the tropical America. These are the some of the exotic plant, showing some exotic plant. But this Iconia crassipes, it was introduced from Britain by a lady during 1914 to 1916. Now it is dangerous for the aquatic system. according to different categories of ICN category. Thank you. Thank you for organizer for inviting me to deliver this lecture, and Bhattacharya and Prashant and also the Devnath. Thank you. This is all.
Thank you so much, sir. Actually, I'm listening to your lecture almost after 11 years, and nothing has changed. Still, you enriched us with lot of information in a really small amount of time, and let us enjoy the cream of taxonomy. Actually, this almost 115 minutes of lecture was enough to cover many parts of undergraduate and postgraduate syllabus of botany. So I would like to ask the participants present here if they have any query. Actually, I have a question.
Yes, okay.
My question is, sir, you said that the codon bonplandianum has changed to codon bonplandianas.
Yes.
Is there any specific reason to change the name, sir?
According to nomenclature concept, new nomenclature advice, it has been changed. What are the exact reason, I don't know.
Okay.
But it is bonplandianas, but previously written in most of the books as bonplandianum.
Yes, sir. Actually, you ended with is like a neuter ending, right, sir? But US is, I guess, masculine, so that's why I think that why is it
I
changed?
don't know the exact rules nomenclature at present. What are the reason for changing the name, this name?
Okay. Thank you, sir. Thank you, sir. Anyone have any question from the participant? As I said, you have covered a lot of topics in very short amount of time.
Okay. Thank you all for your kind patience.
Thank you, sir. Thank you, sir. Thank you.
Thank you. Now I am leaving.
Thank you, sir.
Hello?
Thank you, sir.
Okay.
Thank you very much, sir. We have got a lot of information from your nice deliberation and the lecture, and our students are much enriched with their knowledge, and they are refreshed right now, getting them more and more information, starting from the general reading purpose up to the research. So teaching as well as research purpose in the both things, the diversity and the phytodiversitical work as well as the information, everything they are getting knowledge from your more elaborative and nice presentation and everything, and very nicely, because the epiphytes, parasites, aquatic plants, covering all aspects, they are very much enriched, I think. Thank you very much, sir.
Okay.
For sharing your knowledge with us, and you have contributed your valuable time with us. That's why we are so much grateful upon you, sir. Having your very busy schedule in teaching aspects, but in spite of that, we have got you, and that's why we are very much grateful to you, sir.
Okay. Okay.
স্যার ভালো থাকুন, এরকম ভাবেই যেন আমরা আপনাকে পাই।
এটা নিশ্চয়ই, এতে কোনো অসুবিধা কি আছে? যতদিন অ্যাক্টিভ থাকবো ততদিনই।
হ্যাঁ স্যার
এখন তো রোজই অ্যাসাইনমেন্ট, প্রায় রোজই, প্রায় আজকে এখানকার সেখানে এক এক জায়গায় অ্যাসাইনমেন্ট লেগেই আছে সব। হ্যাঁ, ঠিক আছে, খুব ভালো হচ্ছে, করো।
Thank you, sir. Thank you, sir.
ঠিক আছে।
I guess Miss Rita Ghosh has some question. Rajiv.
হ্যাঁ, বলো।
হ্যাঁ স্যার, বললাম স্যার ভালো থাকবেন, সাবধানে থাকবেন।
হ্যাঁ, সে তো থাকতেই হবে। এখন তো ভালো থাকাটাই হচ্ছে সব থেকে বড় সমস্যা। মানে লাইফের এখন বড় সমস্যা হচ্ছে ভালো থাকা। ভালো থাকলেই সবকিছু চলবে। হ্যাঁ, ঠিক আছে।
ভালো থাকবেন স্যার, প্রণাম নেবেন।
অশোক, অনিশ, সবাই, দেবনাথের সঙ্গে আজকে সকালে কথা হয়েছে। হ্যাঁ, ঠিক আছে। হ্যাঁ, ছেড়ে যাচ্ছি তাহলে। তোমাদের নেক্সট স্পিকারকে তোমরা ডাকতে পারবে। হ্যাঁ। ঠিক আছে, আমি লিভ করে যাচ্ছি।
ওকে স্যার, থ্যাংক ইউ স্যার।
ওকে।
Thank you so much, sir. And with this, I would like to request our honorable convener and head of the department, Dr. Ashok Bhattacharya, sir, to please summarize the outcome for the day 2 of the webinar. Sir, please.
Okay, thank you, Anish, and very good afternoon to all the respected participants and my colleagues present over here. And before summarizing, I'm requesting to all the delegates and the participants right now to announce, although the announcement on behalf of the organizing committee has already been done, but again I'm giving a reminder that due to a sudden accident for one of our eminent invited speaker, Dr. Sandeep Kumar Behera of CSIR National Botanical Research Institute, Lucknow, the schedule of this lecture has been rescheduled and it is shifted from today to tomorrow, and that's why the reschedule of this lecture it will be displayed and it will be communicated to all the participants to join accordingly to be held on tomorrow, and it will start at 10 o'clock. And tomorrow again I'm requesting to all the participants and delegates to spare some time and to consider us for such a mishap, because we are bound to reschedule the program from today to tomorrow. Just tomorrow 1.5 hours we need your patience and time and help and cooperation in all aspects. And tomorrow from 10 to 11:30 the program will be conducted. From 10 to 11 the delegate speech, that means the Dr. Sandeep Kumar Behera, he will talk, and today he is absent due to the sudden demise of his father-in-law, that's why. And we have shifted the program, and after that the prize distribution ceremony and the results of the competition, e-poster competition, will be announced in valedictory session to be held on tomorrow. And at that time our respected judges will also remain present, so that the participants and the e-poster contestants, they can be acclimatized and they can get some introduction with the respected judges tomorrow. That's why I'm requesting to all of the participants and delegates to kindly present tomorrow at 10 o'clock up to 11:30, so that our delayed technical session as well as the valedictory session could be finished accordingly. And under this circumstance, the feedback form will also be given tomorrow in the valedictory session before the valedictory session, so that all the participants can get their online certificate for participation as well as presentation as it is and where it is. And that's why again I'm requesting for your kind cooperation for this mishap. Okay, thank you very much. Now I am summarizing for the today's lecture. Today the poster session started accordingly, and the first contestant Suhani Parekh, she was absent, and that's why we followed the later 6 contestant, that means not the 6, actually the 5 contestants, that was the Shromana Ghosh, Sandeepa Saha, Shampa Kundu, Amije Bhat, and Rituparna Saha. And the last one was the Moutusi Sen, our colleagues, and she was not the contestant, but she presented her work with us and shared her knowledge, and enriched us our knowledge. So the Shromana Ghosh, she presented very nice the particular topic, eucalyptus pollen allergy, the current scenario. Then Sandeepa Saha, she presented flowering plant diversity of Biharinath Hill in Bankura district, and very well, the work is very well, and she presented very well. Shampa Kundu also presented very well, and it is a very compact work, considering all aspects. That was the first Cenozoic macrofossils evidence of the Polypodiaceae from India and its biogeographic implications. Very good work. Then the Ami J. Bhatt, it was also very good work on the Asteraceae from the study of the comparative anatomical study of the Asteraceae member from the Banaskantha district. Then the Rituparna Saha, she also emphasized some ideas regarding the topophagus, that is the first record from India. So this was the poster sessions that we got, and we got so many informations from the contributor in these sessions. Then we followed the next sessions, that is the technical plenary sessions, and the plenary sessions one, that was the plenary sessions, it was the plenary sessions of today's lecture, that was the plenary speech number 3. It was given by our respected madam Dr. Ashalota D. Rosario, where she emphasized on various aspects of the gymnosperm and the paleobotany, and we enriched, and our students also get high and the enrichment knowledge, very recent knowledges, and the interest and the encouragement also for gymnosperm and the paleobotany teaching, as well as some clues for further work, so that this dead subject now can be revived again. And that's why this speech provided by by our respected madam Dr. Ashalota D. Rosario, it is very nice, and I thank on behalf of the Department of Botany to madam. Then the plenary speech 4, it was given by our eminent teacher, Professor Shobhan Kumar Mukherjee of the University of Kolani, and he elaborated the different phytodiversical point of view, starting from the parasitic diversity, epiphytic diversity, aquatic plant diversity, and the many other plant diversity, some nomenclatural aspects. Everything he has covered, and he has given a lot of idea and the informations, and he has shared the existing as well as the current knowledges with us to give a brief scenario about and the encouraging attitude towards the plant taxonomy and systematic research. So, and after that, our program is now finished, and the plenary speech 5, Dr. Sandeep Kumar Behera, he will deliver his lecture tomorrow at 10 o'clock. And thank you to all the participants and the delegates present over here for keeping their patience and their and cooperation, extending their hands for cooperations towards us always, as such, to finish the program very smoothly. And I am very much personally thankful to the to our members, colleagues, particularly the Mr. Anish Bhattacharya, Dr. Sandeep Andre, for conducting the program and the event very smoothly. Not only that, our colleagues, Dr. Pushanto Saha, Dr. Moutushi Sen, they are also there. All of us are trying our level best to complete the event very smoothly. So thank you all, and again we will be meeting tomorrow at 10 o'clock. Thank you very much.
Thank you so much, sir, for your valuable talk. So this concludes this webinar for today. I would like to request the participants to present tomorrow and make the last day successful. So see you tomorrow. Thank you.